Сьогодні у нас відбудеться цикл публічних лекцій. Це так звана така прес-конференція до великої конференції, яка планується у Львові наступного року, 26-й воркшоп Вітенберга з дослідження ефективного ядра. І сьогодні ми зібрали спікерів, членів організаційного комітету для того, щоб презентувати наше місто, презентувати науку світового рівня. Мені дуже приємно, що окрім того, що тут в залі дуже багато молодих облич, молодих науковців, які прийшли, у нас ведеться відеотрансляція. Відеотрансляція цієї конференції, окрім науковців Львова, до нас приєдналися науковці таких давніх українських міст, як Париж, Детройт, Упсала, Чикаго. Я їх також вітаю. Це дуже класно, тому що Львів якийсь сьогодні такий для нас, українців, важливий день, як День прапора, долучився до світової науки. Лекції будуть українською і англійською мовою, більшість англійською, тому що у нас є іноземні лектори. І можна буде задавати запитання, але ми трошки є стримані, ми маємо досить такий проміжок часу для нашої конференції, тому я би просив, щоб це було досить конкретно. Я також прошу виключити мобільні телефони, для того, щоб не заважати нашим докторам. Ну і, напевно, починаємо. With great pleasure, I would like to introduce first speaker, Yegor Vasetsky, head of the Chromatin Dynamics and Chromatin Laboratory from Institute with Overseas France. Please.
Thank you. It's my great pleasure to be here. It's not the first time, not the last time, hopefully. And what you're seeing here is just a preview of a big meeting, which will be held here in next June, and where all of you, those who are here in this conference hall, and those who are listening to me at a distance, will be able not only to listen to lectures, but to participate, to give talks and present their data. I will talk about this later in the meeting to those who will stay with us till the end. Okay, now the topic of my lecture, which is intentionally very popular, is about coronavirus. I mean, everyone is now a specialist in coronavirus. I'm probably a little bit more because we published colleagues paper on how coronavirus enters the cell, so I know a little bit about the physiology. So I'm going to talk about the viral genome and vaccines and how the vaccines are made. And I will try to talk about some myths which surround vaccines and coronavirus. Okay, so coronavirus is, oops, no. Yeah, okay. Coronavirus is not a new virus. It belongs to a well-established family of coronaviruses, which are basically all the same. They have several proteins, but this is actually medium-sized virus, but it has a very, very large RNA genome, which is about 30 kilobase. It has a single-stranded RNA inside. Outside, it has the famous spike protein, which everyone knows about, which is fixed on a nucleocapsid protein. There is also a membrane-like protein. So it's quite an easy structure. well, the first myth is that actually the coronavirus was linked to Donald Trump. No, it's not. And actually, the theory that Donald Trump named coronavirus came from Russia's Channel 1. It's not true. Well, the genome encodes 5 structural proteins: spike, which is outside, M, membrane protein, N, nucleocapsid protein, envelope protein, and if you're interested in to see how coronavirus enters the cell and replicates, there are resources which are listed here, which you can just see. What is important is that actually coronavirus uses its spike protein to bind to the cell membrane receptor, which is called ACE2, angiotensin converting enzyme, which is absolutely necessary for the function of many cell types. And this fixation allows it to enter the cell. ACE2 receptors are present on many cell types in the lungs, in the mouth. It also can infect heart cells and other type of cells. And so it uses a natural receptor to act. Okay, now let's go to the vaccination and to understand how vaccination works. We need to understand actually how the adaptive immunity works. Basically, when the pathogen is detected, macrophages eat pathogen, then parts of the pathogen appears on the surface of the macrophages. They are bound to T lymphocytes, and then T lymphocytes transmit these parts of the pathogen to B lymphocytes. B lymphocytes actually, they already have an affinity to, slight affinity to different types of pathogens, and those B cells that recognize the pathogen, they they get activated. They go to the lymph nodes where they replicate and where they get better affinity to the pathogen, and then they start producing antibodies which recognize the pathogen, and this actually protects the organism. After the infection, what happens actually, you have lots of antibodies and lots of B cells. But as we get infected every day, we cannot keep large amount of B cells recognizing a specific pathogen. So with time, they just disappear. But what remains are memory B cells. Memory B cells are cells that persist. The specific cells that recognize the pathogen, they persist, and if the pathogen infects again, now the cell has the abdomen has these cells which immediately recognize the pathogen, and as they recognize the pathogen, they start to proliferate, and the immune response is almost immediate because it takes the organism several days. We could do to develop efficient antibodies upon primary infection. Okay, now how do vaccines work? As I said, we have a repertoire of existing antibodies against different diseases, and when we get reinfected, because what happens actually, the immunity does not protect from infection. It allows to fight infection rapidly so that you don't notice it. So you get the antibody, and the antibody recognizes the pathogen. The pathogen is easily eliminated. Now, when there is no antibody, you get infected, you get a disease. And the vaccine actually works by taking a part of pathogen, of new pathogen, to whom you don't have immunity, and introducing it to the body. And this kind of emulates the infection without the negative consequences. Well, I cannot say that there are no negative consequences from immunization, but they are far far less than the consequences of the infection. And you also know that the consequences of COVID infection can be really very, very bad. Okay, and the infection actually, the vaccination actually not only protects the person, but when a certain level of vaccination is attained, we get what is called herd immunity. If like 70%, 70 to 80% of the population is vaccinated, the virus is not transmitted, no more transmitted from individual to individual. That's actually how it works, because we don't have poliomielitis, we don't have pox and other diseases which were really serious in the 19th century, and now they are almost eliminated because of the herd immunity. Now, how vaccines are developed? As I said, we take part of the antigen and introduce it into the body. In order to enhance the immune response, sometimes we add the adjuvant, which increases the immune response. Sometimes there are preservatives, stabilizers. In order to get the molecules into the cells, there are surfactants that make a kind of mini cell and protect the content. This is particularly important in the case of RNA vaccines. And so all these components are added to the antigen and are injected into an individual. Then what happens when a vaccine is developed? It undergoes 3 phases of clinical trials. First phase usually is used to test the safety of the vaccine, to see whether there are any negative consequences. Phase 2 trial carried out on a relatively small amount of people tests the efficacy of the vaccine. And phase 3 is carried out in case of vaccines on several hundred, tens of thousands of people, and it measures whether it really protects and whether there are negative effects. Because vaccine is a very dangerous substance, because unlike drugs, for example, which can be administered to few people, vaccines are administered to millions of people, and probably in the case of COVID, probably to billions. So even smallest negative consequences will be immediately visible. So it has to be, the vaccine has to be really safe and really efficient to be able to play its role. Okay, so what are the ways to make a vaccine? There are 3 major ways. You can use a whole virus or a bacteria, if it's bacterial infection, to introduce it into the body, and you usually either kill a virus or bacteria before introducing it, or make it or weaken it so that it cannot cause negative consequences. You can use parts of the virus, protein parts of the virus, or you can use the genetic material of the virus. Before COVID era, these 2 approaches were predominant, and the nucleic acid vaccines were only used in very rare cases, for example, in vaccination against cancer. Now, let's briefly consider different types of COVID-2 vaccines. The whole microbe was the first developed. So you inactivate the virus, usually using formaldehyde or other cross-linking compounds, so it's no longer dangerous. And you inject this virus, the virus is dead, but it still causes the immune reaction of the organism. And examples of these vaccines are Sinovac or Coronavac produced in China, or Covivac, which is produced in Russia. The advantage of these vaccines is that they are relatively easy to make. They're quite stable, but the immune response is not well. The immune response is not very good. It usually protects about 60% of people from reinfection. The next approach is the civilian approach. So you take the protein or peptide and inject it to the body, which is supposed to make the antibodies. And the only known and used vaccine is Epivac Corona, which is produced in Russia, and this is a huge failure because it doesn't protect at all, for reasons that I don't have time to discuss. And now the genetic approach, which is quite interesting because it has never been used in large scale immunization before. So what you do actually, you use the genetic material, it can be DNA or RNA, to introduce it to the cells. Then the proteins will be synthesized in DNA or RNA, and then the body will recognize these proteins. In case of these COVID vaccines, this is the spike protein or parts of the spike protein, and it will develop an immune response. The first vaccines developed using this response used another virus, which is called the adenovirus, which affects human cells. But instead of adenoviral DNA, the scientists put a part of DNA that would synthesize parts of the spike protein, and this virus, upon infecting cells, would not replicate because it does not contain the sequences which allow the virus to replicate, but it would produce the spike protein. These vaccines are produced by AstraZeneca, Johnson & Johnson, and there is also a Russian version which was Sputnik V or Sputnik-like, and they differ by the types of adenovirus used, and for example, AstraZeneca uses the adenovirus from chimpanzee, Sputnik, and Sputnik uses Johnson & Johnson uses human adenovirus. Now, the last type of vaccines, which are now popular, are produced by Pfizer and Moderna. These are vaccines that only contain RNA, which is the basic molecule which is necessary for protein synthesis. This RNA is injected into the body. It is included into kind of mini cells, lipid membranes, which fuse with cellular membrane. The RNA gets into the body, gets translated, the protein is produced, and it is synthesized. Okay. Now, I don't have a lot of time, so I will probably keep some time for questions. Now, some practical things. So what can you expect before, during, and after COVID vaccination? Well, before, actually, you should choose the vaccine if you have the chance to do it, and probably ask whether vaccination is recommended. Then, during vaccination, read what could be a negative consequences, and after, you could expect some side effects. I had some side effects after vaccination. People from my laboratory who were injected with AstraZeneca vaccine were all sick for a couple of days. But then you should realize that vaccination does not fully protect you from infection. It helps fight infection. So even if you're vaccinated, you should be careful not to get infected, because if you get infected with a large dose of virus, you will still get an infection, which will probably be in a light form, but still be an infection. Okay, so now before stopping, I will just talk about some popular myths. So, do COVID vaccines contain microchips? Unfortunately, no, because we could not. I mean, the microchips which have not been developed yet, small microchips that would do something useful. Will a COVID vaccine alter DNA? No. There could be cases of integration of DNA into cells, but they are very improbable, and actually they will not alter, definitely not alter the DNA of the germ cells. So this, and we should also note that every day, even now, there is integration of foreign DNA into one of the cells in your body, just now, but it doesn't have negative effects overall. Now, is it COVID vaccines is in pregnancy? COVID vaccine does not prevent someone getting pregnant, and it protects people, because actually, if you get infected during pregnancy, some of the drugs which are used to treat COVID-19 are not recommended during pregnancy. So the choice of treatment is very much reduced if you get infected. So of course, the COVID-19 vaccine cannot infect you with COVID, because it uses either dead viruses or parts of the virus. And finally, if you get the COVID-19 vaccine, you will not test positive for COVID-19 on a viral test, on the PCR test, or on a antigenic test, because antigenic test usually uses a couple of viral proteins for the test. So I'll stop now, and we'll be happy to take questions.
Questions. First, as coronavirus survivor, I would like to ask you, you didn't mention any brain cells, any neurons in your list of coronavirus affected cells.
They are, of course, they are infected.
So
yeah,
I expect some consequences, right?
Yes.
Okay.
Actually, there are
one brain damage.
Yeah, yes, it's true, because in the case of severe coronavirus infection, there are consequences for the brain, and people recover from them, but they learn with more difficulties. Another reason to get vaccinated.
And the second question is about peptide vaccines in general. So because the failure is more with design of peptide vaccines rather than with theory, right? That they selected the peptides linked to the internal protein of virus. But my question. We don't know other peptide vaccines, right?
No.
Somehow they're not working for other infections as well.
Well, the same company tried to produce a vaccine against Zika virus, but they were not very efficient either. Well, proteins are very efficient, and actually most of the vaccines which are used to treat kids are protein vaccines. But the peptides, actually there is a question of folding, because all the proteins are folded, they form a kind of 3D structure, and I'm not sure that the peptides can be folded the same way. As a researcher, we all work with antibodies, and we know that antibodies produced against part of the protein, in many cases, they do not recognize correctly the protein which is in a cell.
I have two questions. First question, okay, most of the vaccine, all of the vaccines are made against this S protein, right?
Yes.
Do you think there may be other proteins which may serve as a target for new vaccines, which may work together, for example? That's my first question. And the second question, do you know if a drug will be out there in soon or in any foreseen future, which may help to fight COVID?
Well, for the drugs, there are some drugs that are somehow efficient.
But they are not specific.
No, they're not, definitely, they're not specific.
I
And
thought
one
it
of
was
the drugs,
specific.
one of the drugs is a drug which is called famotidine, which is a drug against the indigestion, which you can buy in pharmacy, and it works on first stages of infection because it blocks one of the enzymes of coronavirus. It's not very efficient, but Trump, when he was sick, was treated with this drug. Then the question about the other proteins. Yes, it's true. Some patients, and we can get insight about this from the natural antibodies which are produced by coronavirus infection, and some patients develop the antibodies against the capsid protein, the M protein. But there is data that these people, those people who get the most antibodies against the N protein, they get reinfected more than those who have the antibodies against the N and S and other proteins. And then there is also another logic in this, because the capsid protein, okay, it can mutate, okay, but the S protein is necessary to get into the body, into the cell. So the extent to which it can mutate is limited. It does mutate, of course, because all these variants, delta variant, etc., they mutate, but still the parts of the spike protein remain conservative.
Very short one. First, thank you very much for this wonderful overview. It's really excellent that you've combined together all what we know about vaccine and virus. Thank you. My short question, that's a little bit off topic. There are some publications about integration of virus in the human genome. So what is your opinion on this, and how would it change our future view on vaccination and treatment of disease, as well as PCR diagnostics that would give us, if you wish, false positive every time for those people?
Safety.
Yeah.
Safety.
As far as I know, there are problems with the first publication. It was retracted. Yeah,
Okay.
yeah, and but of course, I mean, any virus, any genetic material that gets into the body can potentially integrate. The fact that it integrates into somatic cells does not pose potential danger to the, well, it has, every integration is a potential threat to the body, because it can integrate in a strategic place, it can cause mutations and everything, but the probability of this is very low. So for me, I wouldn't expect much. And the example this is the Epstein-Barr virus, which is DNA virus, which infects 99%, 90% of the human population. It is present in all of you. And still, there is a very few integration into human cells. So for
Okay, so it's moving target, right? Okay.
Thank you.
so
Thank you, Eva. For this presentation, I would like to invite my speaker, Nick Butler from University of Leicester.
Thank you. First of all, I want to say thank you very much for kind invitation to this wonderful city. I had a chance yesterday to move around. This is a fantastic architecture, a very interesting mix of different cultures. And I hope that I will be able to come next year and enjoy my time here. Okay, today I will talk about a little bit about P53 because my labs are working on this very interesting protein. P53 is supposed to be, is considered as one of the most important tumor suppressors in in human body. So in response to different kind of stress, that could be oxidative stress, hypoxia, damage, oncogenes, and even viral integration. P53 is activated and it participates in different signaling programs such as senescence, angiogenesis, autophagy, cell cycle arrest, apoptosis, and metabolism. So the question is, how does one protein can do so many things? Actually, in addition, P53 is known to suppress metastasis. So this is a simple experiment where we take immune suppressed mice inject with lymphoma cells, which express GFP. So you can see green color. Tumor cells accumulate in lymph nodes, where they're supposed to be, in case of P53 wild type. And in the animal where P53 is absent, you see multiple metastases in lungs and even brain. So P53 is important. Now, the question is, how does it do it? It turns out that P53, as many tumor suppressors and oncogenes, works as a transcription factor. What does it mean? If in response to stress, P53 binds DNA promoters of its target genes, whose products either induce cell cycle arrest and cells survive, or they induce cell death, and they will die. I can give you an analogy of P53 as a bomb with a clock, right? So at a certain time, clock ticks, boom, you get an explosion, cell dies. Now, in general, you don't want to have a bomb in your house because, God forbid, something goes wrong, your house gets exploded, right? That's why P53 is always under constant check by different enzymes, such as WIP1 phosphatase. I hope we will hear more today about this wonderful enzyme. Also, MD2 is a E3 ubiquitin ligase, which ubiquitinates P53, and that causes its degradation in proteasome. Also, I will have no time today to talk about other targets of P53, such as microRNA and non-coding RNAs, but you may ask questions later. Now, talking about P53 binding to DNA, you have to remember that in the nucleus, there is no naked DNA. It's all compacted into protein. Now, the basic unit of protein is a nucleosome, which consists of histone monomer, H3H4, H2AH2B, which are wrapped around with 146 base pairs of DNA. And histones have long, unstructured tails, which are modified by different enzymes. What is important to remember is that nucleosome can slide along DNA, opening some spaces on DNA, and that's where transcription factors bind. And histone modifications are very important for this process. Now, if you look at the sequences of histone H3 and the C-terminus of P53, you will be amazed how many common residues you see. And of course, we know that histones are subject to multiple modifications, and we wondered, and other laboratories also have the same question, whether P53 can undergo the same modifications as histones by the same enzymes. And the answer to this, yes. So this is a sequence of H3. You see that lysine residues can be acetylated, methylated, ubiquitinated, and many other modifications. This is a slide showing the work of many laboratories. The most prominent of them is Lady Brewer's laboratories, Bob Rader, Schellenberger, also Lee made some contribution into that field. This is the structure of P53, and you can see that in various places, P53 can be acetylated, methylated, ubiquitinated, and they all have different functions. What was interesting is that depending on the site of modification, acetylation can either induce acetylation of P53 can either induce cell cycle arrest. So that means that genes which activate cell cycle would be expressed preferentially, or if acetylation occurs in the binding domain, that would induce apoptosis. So you see that the same modification, depending on the site, can do different effects on P53. Another thing you have to remember is that P53 modifications in P53 are not hectic. They occur in a very coordinated manner. I would say sequential order. So, for example, we have shown that P53 undergoes methylation on lysine 372, and that induces its subsequent acetylation on another side, lysine 382. This is a simple Western blot where we introduced either mutant P53, which cannot be methylated, or wild type P53, which can be acetylated. And you can see that the amount of P53 with the absence of methylation and acetylation goes down. So clearly, this is important for the amount of P53 in the cell. Then we'll get again, depending on the site of methylation, very similar to acetylation. Methylation can either induce P53 function or repress P53 function. There are several papers showing that, but I don't have time to really go through this. Just to round up the first part of my talk, I will show you this cartoon. So you have P53 in binds MDM2, which is an E3 ubiquitin ligase. It ubiquitinates the C terminus of P53, which otherwise targets p53 for proteasome degradation. But in response to gain damage, genotoxic stress, p53 becomes phosphorylated, methylated, acetylated, becomes stable, and that means it can activate the transcription of its target genes. After the stress is gone, I have to say that both GRIP1 and MDF2 are the targets of p53. So p53 at the same time sort of promotes its own death. After the stress is gone, p53 will also disappear because GRIP1 will probably dephosphorylate p53, another enzyme HDAC1/2, which are deacetylases. They will deacetylate p53, and then p53 gets ubiquitinated and goes to the proteasome. Okay. So after the first part of my talk, you would say, okay, it's great. So post-translational modifications are important for p53. And the second thought you may have is that, okay, it would be great if you somehow break the interaction between p53 and MTM2, right? Because in this way, you will activate p53, and p53 will supposedly will kill the cancer cell. That's what we want. Now, in fact, in 2003, 2004, Lubo Vasilyev in New Jersey, he synthesized a molecule which exactly does its job by breaking down p53 from MDM2. The way how it works is this molecule binds the N-terminus of MDM2. So I have to say that p53 binds MDM2 with its N-terminus and with hydrophobic residues into this pocket of MDM2. And the small molecule mimics the structure of p53. So this is a this is a small molecule which inhibits MDM2 binding to p53. Having this hypothesis in mind, we searched for our own inhibitor of MDM2. And Pavel Davidovich helped to do the bioinformatics screen. We analyzed about 500,000, it's a mistake, 500,000 molecules in the virtual screen and came down with 100 compounds which could work as inhibitors of MDM2. Then we tested them all using cell-based system, and we came up with 6 hits which worked in cells. The test whether this how much time? 0.
No,
Okay.
just continue.
I need 2 minutes, if possible.
Yes.
Sorry. So the test system was very simple. You get GFP under control of p53 response elements. So if p53 is activated, it binds the promoter of this plasmid and activates expression of GFP, and you get green cells. So I'll skip that. What is interesting is that our compound was able to activate p53 and induce cell death in various tumors. For example, in colon cancer or osteosarcoma, and it was able to induce cell death in p53 positive tumors. That's important. And also it helped to kill tumors, not kill, but promote the survival of animals with xenoblasts. Now, having told you this, unfortunately, all the clinical trials so far on activation of p53 failed. None of the molecules which activate p53 became a drug. unfortunately. Why is that? There are two reasons for this. One is inactivation of the apoptotic pathway, which is downstream of p53, and another one is mutation in the p53 gene. So just to remind you, there is extrinsic pathway of apoptosis, and this is the intrinsic apoptotic pathway. p53 induced restriction of vaccine part, which likes channels in mitochondria, and this induces cytochrome C release, and that results in activation of apoptosis, which then induces caspase pathway. So one way would be to overexpress BCL2, which counteracts BAX and BAT, and another way is caspase 3 inactivation, which is important for mediation of cell death. Another way to fight the activation of p53 pathway would be through mutations in the p53 gene, because the structure of the p53 gene is made in such a way that there are naked DNA in the gene. So mutations would cause an opposite effect on the cell. So just to finish with this, so the good p53 is a tumor suppressor. It induces apoptosis, immune system activation, prevents metastasis, but inducing p53 inhibits apoptosis, it induces genomic instability, so it fights p53. So to conclude, I would say that now there are several clinical trials in combination with different drugs. One of them is venetoclax, very interesting. Some of them are inhibitors of immune checkpoints. And I'll talk more if you want, but I have to finish. With this, I will be happy to take questions. Thank you.
Okay, thanks for the nice talk. My introduction of my favorite target protein, it's DM1B. I have two questions regarding the therapy. The first question, do you know the reverse situation, successful drug that inhibits p53? Because in many situations, like for example, the disaster, I remember Chernobyl, Japanese nuclear plant, it's important to suppress the function of active radiation poisoning.
The quick answer to this very long question is no.
Okay. So the same situation like with therapies that activate p53 wild type. And do you
Very
think
ambiguous.
that we probably need to drop this thematic of restoring wild type function and just focus on using this specific feature, because we think that about 70% of tumor has mutation in great mutation in p53. Another 20 probably has some negative activators like MDM2 with one mutation that activate them, and this is a way how to suppress p53 function. Maybe we need to use this feature and differentiate ourselves from normal cells. And
Yes, I would rather focus on those 20% where p53 is active. As I showed you here, that activation of p53 on its own is not enough, right? Because you get mutations in the apoptotic pathway, you can get BCL2 activation, you can get MDR1, which is a pump which gets rid of the drugs. In combination with all these drugs, maybe we can get a synergistic effect, hitting cancer cells from different sides, from the point of view from immune evasion, point of view, metabolism, and other features. Yes. Yes.
What about the interrelations with PARP and all these issues? So
Yes,
it appears that
Yeah, P53 activates PARP, and PARP is a cleavage of PARP is very important for apoptotic pathway. And in fact, when P53 is activated, it has cleavage of PARP, and this will result in induction of the apoptotic program.
Unfortunately, we don't have time for more
Yes,
questions,
thank you.
so thank you, and you can ask questions to me. And please keep to the time, because we are already quite
Anti-mitotic chemotherapy resistance and great pleasure. I would like to invite Alexander Ishel from University of Florida, United States. Sorry.
Great. Good morning, everybody. For participation, and I want to use this opportunity to thank organizers to make chance for all of us to come here to enjoy our scientific discussion and also to enjoy the beautiful city of Lviv. It's my second visit here. First one was exactly 40 years ago, and the city is getting better and better and better. Well, I don't think that I will be able to visit in another 40 years, but next year indeed I'm looking forward to this. So what I will try to talk to you in next 18 or 19 minutes is one of the topics of my laboratory, which is anti-mitotic chemotherapy resistant. And specifically, we will be talking about two aspects here, who is to blame and what to do with this anti-mitotic chemotherapy resistant. That's our line of talk, and we will start with anti-mitotic therapy in cancer. So in this regard, we are focusing mostly on two types of cancer, which is prostate cancer and breast cancer. And the reason is that those two most devastating cancers in the United States and actually in the Western world. And that's all these numbers that you see here. And when we are talking about cancer chemotherapy, indeed, you know that there are some targeted therapies. And here on the left, you see this very short list, definitely incomplete list of targeted therapy, where first few are towards breast cancer, the last one is towards prostate cancer. In addition, we shouldn't forget that there are some general anti-cancer drugs that are just cytotoxic, and they act as either DNA damage drugs, and that's the examples here, or so-called anti-mitotics. And those anti-mitotics, here we will focus on the drug which is called Taxol or paclitaxel. It's quite an interesting story by itself, because this drug was introduced in clinic in 1983, and 20 years later, it's won this national historic chemical landmark because of the enormous, wonderful success in the clinic. And what is the mechanism of activity? Taxanes binds to microtubules, and it inhibits depolymerization of microtubules. So those microtubules, they became just stuck somewhere in the process. And as we know, dynamics of microtubules is very important in interface cells and indeed in mitotic cells. So as in clinics, major limitations of taxanes are toxicities, which lead to the novel formulation of taxanes, and resistance. So when patients are first treated with taxanes, the level of response is about 50%. And when patients are coming to the second round of chemotherapy treatment, this resistance came up to the 70 or 80%. And now imagine with the huge level of side effects. The positive of the first treatment is just 50%. On the second line of treatment, it drops several times down. So what are the reasons of this resistance? Mutations in tubulin, the direct target of toxicity, are quite rare. And the main notion in the field of this resistance, which come from the effects in the mitotic checkpoints. So now, indeed, goals of number of laboratories, including our laboratory, is to identify predictive markers of this resistance and understand the mechanism of anti-mitotic drug resistance. Here is a picture of wonderful orchestrated process that we know as mitosis. You saw it probably already in kindergarten, just a reminder. And that's the most important when we go from here in here, from metaphase to anaphase. What happens? You have to divide equally, precisely, genetic material between two daughter cells. And how it's done? That's our microtubules. Each of them will be connected to one of the daughter chromatids, and one time would come very synchronously. These chromatids would divide into these two daughter cells. And now, for these microtubules to get proper attachment to kinetochores, you need a very complicated regulatory mechanism, because the connection between microtubules and kinetochores is a very stochastic process. You can imagine that when microtubes start to grow up and try to hook on these kinetochores, you can have all type of events over there. Somehow they are corrected till both chromatids are properly attached, and then something happens, and boom, we go from anaphase to telomerase. Now, that's illustration of this process done on the microscope. And here you have several markers of mitosis, and here is transition from metaphase to anaphase. What happens when we add toxins to the cells? They enter mitosis, okay, and then they stay, and they stay, and they stay, and then they get tired, and they exit. But they do not exit in these beautiful daughter cells. They exit as this mitotic catastrophe. So the genetic material is not divided between daughter cells, it just collapses, which is called mitotic catastrophe, which, as you can see, one can recognize easily on the microscope with this abnormally shaped nuclei. So now, if you take this knowledge and come to the anti-mitotic treatment, what we can generalize that after you add these drugs in the cells, or when patients are treated, so you have this block, and then sensitive cells will undergo mitotic catastrophe, and then different type of cell death. With the resistant cells, they have enough courage to stay in this block longer. And now in our cell culture models, if we wash toxins away, within very few minutes, dynamics of microtubules will be restored. Cells can continue this division and they can survive. Same happens in patients. So basically, resistant cells, they can stay in mitosis longer and keep this block to be divided after decay of drug. And here I will show you just very brief vignette of the wonderful work which was done by the two former graduate students, Phosphoryls and Serena Gymnasi. It is actually one slide that combined together three publications they identified two molecules that are important in mitosis. These molecules are called DAX and USP7. And with cells where DAX or USP7 are mutated or reduced, you have accumulation of the very important mitotic protein Aurora A, which will promote accumulation of cells in this mitotic block. It will result in this resistance, in addition, in genomic instability. And they came with these several wonderful conclusions that can be translated into clinics, that we shouldn't use, we can use DAX and USP7 as predictive markers for taxane response. And we can use Aurora A inhibitors, which are clinical trials with taxanes to improve activity of taxanes. And we shouldn't use these USP7 inhibitors in combination with taxanes. And I must say that USP7 inhibitors were promoted by several pharmacological companies because of the connection between USP7 and P53. That, as we know, based on the previous talk, is very prominent target. And now, what shall we do with this? Okay, we identify those markers, we know the mechanism. But how we can move from here into here? And here I will tell you a couple of interesting and brief stories. And the first story is that we should keep our eyes open as experimental biologists. So this former student, Serena, she did once an experiment, absolutely unrelated experiment, where she tried to use heat shock, just hypothermia, 41, 42 centigrade. Was completely unrelated project. And then she decided to see what will happen if you combine taxanes with this hypothermia. And she ran into my office screaming, Alexander, you must go to the microscope and see what's going on over there. So here is control cells, and here you see DNA of control cells, few cells in mitosis, lots of cells in interface. You add taxane, cells are in mitosis. And now, if you put cells at 41 centigrade, just 1 hour or 2 hours, all cells will go into mitotic catastrophe. So, moreover, she did this wonderful experiment. She compared activity of taxane alone and taxane in combination with heat shock on either control breast cancer cell line or taxa or paclitaxel resistant cell lines. See, they are very resistant. And now, if you combine this heat shock with paclitaxel, you can eliminate much more cells compared to the paclitaxel alone. Great? Hypothermia. Great. But how would you apply this hypothermia? There are different ways to do it. You can do this magnet nanoparticles, and we start a couple of collaborations in this direction. But our idea was to understand the mechanism. And basically here, it's another kind of, I would say, serendipity. Indeed, we were trying to just go systematically after mitotic checkpoint proteins. But then one day I was sitting in my office, drinking coffee and going through the only scientific journal that I keep receiving as the hard copy, which is just a non-journal, which is called Cell. So I'm going through it, and then on one of the figures, I saw something which immediately struck my attention. I saw the picture that reminds me exactly mitotic catastrophe. And I said, what's going on? And indeed, it reminds what you would see when you apply heat shock. So in this paper, they used inhibitors of protein, which is called MPS1. They use small molecule that would inactivate this MPS1, which results in mitotic catastrophe. Now, you saw this picture before. how to control this proper transition, you need so-called spindle assemble checkpoint. Very complicated, several hundred proteins complex. But one of the key molecules that control proper activity of this complex is MPS1. It just, if you wish, traffic light regulator. It will block transition between metaphase to anaphase till the last chromosome will have proper attachment of microtubules. And once all of them are properly attached, it will say, okay, let's go ahead, let's divide. Now, if you fake this MPS1 with inhibitors, what will happen? Cells will come into mitosis and they will leave. They will leave without proper alignment of chromosomes. And indeed, idea was to combine taxanes, in this case, cabazitaxel. That's the last resort of patients, prostate cancer patients, that are coming in the clinic after we already used all the tools as the inhibitors, as surgery, as everything. So that's the last resort. And we wanted to use this cabazitaxel together with MPS1 inhibition. Was a question, and indeed, we opened the literature, and we try to realize that you are not alone, that people developed several MPS inhibitors, and some of them were in clinical trials with breast cancer patients. So now we want to use it in prostate cancer patients. And here there are first encouraging results. We use these 2Z cell cultures, you set cells on plastic treatment, and you clearly see that on number of colonies, which are here on the bottom part, if you combine this treatment, you have much greater reduction, and that's calculation, which shows synergistic effect between these two drugs. And indeed, we wanted to move in 3D culture. That's a wonderful toy, if you wish, because it's not very easy, it's more expensive, but you can brew up the cells in something which would remind metastasis. That's a 3D culture. There are some contacts between the cells, and that's kind of in vivo experiment for poor people. Right? It's much faster. And what you see here, that's those prostospheres with control conditions. We treated with cabazitaxel, we see some reduction. We treated with weight, but now see what happened when you combine this. That's a gray here. It's nothing left here, nothing. If you treat these prostospheres, very first 2 days after you set them up, or 1 week later, the result is about the same. You almost completely eliminate these prostospheres. And now, what you can do also, a number of very interesting tricks with these prostospheres. You can fix it, you can slice it, you can stain it with number of markers, as on this one, for instance, here you see a combination of this KI67, that the marker of cell proliferation, or serine 10 phosphorylation of histone HT, which is marker of mitosis. And again, you combine treatment, you lose all these markers. Whatever is left over, the cells are not proliferating, they are not going through mitosis, that's it. So, and finally, you can collect cells from these prostospheres, resuspended, put it back on plastic, and again, in all other cases, you have plenty of survived cells, but if you combine treatment, it's nothing. Everything is well. So, conclusions from this last part of the presentation is that inhibition of this MPS1 or this mitotic exit, and we expect that this combination can overcome resistance, killing castration-resistant prostate cancer cells, regardless of their sensitivity to cabazitaxel. Also, regardless of different status of androgen receptor, because in this stage of prostate cancer, cells often express truncated or mutated androgen receptor, and we know we have experimental data that our combined treatment works on cells regardless status of androgen receptor. And we also know that we can reduce amount of these taxanes in combined treatment, and ultimately in clinic it should reduce cytotoxicity of cabazitaxel, which is one of the states and aspects of these drugs. Coming back to the title of my presentation, who is to blame? At least in some cases, well, it's my 19 minutes. Almost. In some cases, we should blame prolongated block. And what shall we do? We shall try to force this mitotic exit, and we identify at least 2 ways to do it, with either hypothermia or MPS 1. And that's definitely the most important slide. That's the lab members that did this work, mostly started by Corey and Serena, and now it is continued by new graduate students Xavier. And we have a number of collaborators in the University of Florida and outside of the University of Florida, and indeed grant support, which is in the book. Thank you very much, and I will be happy to answer questions if I have a time. Please.
I don't have a question, but I have one short one. Going to sauna, will it help? Like how much we can keep? How much?
Actually, given an example of a previous speaker, I would say yes. It will.
But did you try?
On yourself.
But on a little bit more serious aspect, I've heard some anecdotal stories that somewhere in Russia, actually either in Novosibirsk or somewhere else, they did some of the experiments of submerging patients in the hot tub while they're transfusing toxins. Now, just to extend it a little bit, in Europe, actually, for the last 10 or 15 years, it is a tendency to reduce level of chemotherapeutic treatments, and old good hypothermia became more and more prominent either by itself or as a complementary aspect. It was known already 50 years ago that centigrade, you increase the temperature, activity of drugs, which increase. I think what is important in our study is that we identify a mechanism, and we also know the sequence. First, you have to transfuse with those drugs, and then cells will be accumulated in mitosis, and then you go to cell. Okay. Thank you for the question.
Thank you.
Thank you, Rick.
Dedicated to the mass spectrometry imaging, great pleasure. I would like to invite Piotr Lido from Marie Curie National Research Institute of Oncology from East Poland.
Thank you. Good morning. Thank you for the opportunity to participate in this wonderful meeting. I was told that cell nucleus is not mandatory on this meeting, so because for the last years my group is more interested in mass spectrometry and proteomics, I would like to introduce a method which is based on mass spectrometry and is more frequently used in different fields of biomedical sciences. Not cell biology yet. I will address this problem at the end. I'm going to talk about pictures, so for me it would be much easier if you can see these pictures. Okay, but nevertheless, the basic principle of mass spectrometry is known from the end of the 19th century. If you have ionized the molecule, you can estimate the mass to charge ratio. If you do it precise enough, you can say, what is this? What was the problem for biology? The majority of biomolecules are fragile, so different methods to ionize just break it into pieces and make it useless for analysis. So development of so-called soft ionization methods was very important. How important? Two guys that developed these methods got Nobel Prize for this discovery. So I think it was important. Okay, so one of the methods developed by this guy, Koichi Tanaka, was so-called matrix-assisted laser desorption ionization mass spectrometry. In this method, we mix our analyte with matrix. Then, after irradiation with light, the analyte gets detached and ionized. The matrix is usually aromatic acid, so because it is aromatic, it can absorb light, and because this is acid, it could generate proton to get our analyte ionized. Usually, MALDI is associated with time of flight type of analyzer. Simply, this is the tube. Small, faster compounds reach to the detector faster than the larger one. And we can see different species here, which are different in the speed to get to the detector. And finally, we see mass spectrum. So on this axis, we have MZ, mass to charge ratio. On this axis, we have intensity. Okay, this is the idea of mass spectrometry, but we are in biology. And also for biologists, this phrase, the picture is worth 1,000 words, is also valid. So in fact, one of the reasons that many of us are in cellular biology is that we generate lovely pictures, right? So the same we can get with mass spectrometry. Simply, instead of analyzing a single sample, we can put whole tissue section to the spectrometer. Then we can get a series of spectra from different pixels just to get co-registered image, which involves molecular image, which is based on this mass spectra, and morphological image. So, for example, we can say for different areas of the tissue in this artificial course, we have heat maps or intensity of different compounds. With this method, we can analyze proteins, lipids, drugs, metabolites, everything which is ionized, and we can analyze what is this based on the mass. Unfortunately, nucleic acids cannot be analyzed by mass spectrometry. This is just the reason of chemical monotony of nucleic acids. Okay, so this is the flow chart. This is slightly more complicated than mass spectrometry, a protocol that we know from chemical labs. Simply, we have to process histological tissue, and also the problem at the end, because in one experiment, we usually register hundreds of thousands of spectra for one specimen, which translates into several gigabytes of data for one tissue specimen. So we really need heavy computation, first to get the components, then to do what we love to do with this type of data, I mean unsupervised image segmentation, just to uncover the hidden molecular structures, which are not visible from the morphological level. Okay, few examples. I'm from Institute of Oncology, so examples will be related to cancer. First, when we do proteomics, usually we want to go into biomarkers. Biomarker mean the compound, which, for example, have different level between cancer and normal tissue, or between different types of tumors, or between different areas in tumor. Okay, so what we can do, these are tissue specimens stained with hematoxylin and eosin. So pathologists would say that this is cancer region, this is normal epithelium, these are muscles, whatever. This is head and neck cancer. Then we can do imaging, and we clearly see the molecular maps that define specific molecules. In this case, these are proteins, but different abundances in different areas in tumor and in normal. But this is the most trivial usage of this method, because, as I said, the nicest part is that we can do unsupervised segmentation, and we can uncover something which is from the morphological level is not visible. So, for example, we can do this clusterization, we can combine this cluster, we can say how it's related to the areas that pathologists see. And finally, we know that different subregions of cancers could be discovered which are not seen by pathologists. This region corresponds, for example, to infiltration of the stroma cells, with the necrotic areas, with different metabolic activities. Okay. Next example, again, biomarkers, we have different components of cells, not only proteins. We know that proteins and nucleic acids are most frequently biomarkers, but also lipids, okay? We have plenty of lipids in cells, so the question is, can we use lipids instead of proteins as a biomarkers? Okay, so what we did, in the same tissue specimen, subsequent sections were used. One of them were used for protein imaging, second was used for lipid imaging, and we can look for these components that differentiate normal tissue from cancer. Okay, we can see that this is this distribution is a distribution of components that are increased in the power to discriminate the cancer and normal tissue. Okay, so definitely there's much more peptides that differentiate cancer tissue from normal tissue. But still, if we are talking about biomarkers, usually these are multi-component classifiers. Okay, so it could happen that even though that single component is not good enough, if we combine many components, we can get a good classifier, which means a good biomarker. So we use different components from this cancer in normal area. We build this type of classifiers. Okay, we found that more or less, even though there is much fewer lipids that discriminate cancer in normal epithelium, still, when we have classifier, multi-component classifier, both lipids and proteins discriminate cancer in normal tissue with a similar amount. Okay, next question. We have metastasis. Okay, usually the problem, the major problem is the primary tumor, but we also have concurrent metastasis to lymph nodes, which defines some both diagnostic and therapeutic problem. We would like to know what is the difference, molecular difference related, for example, to metabolism between primary location and lymph node metastasis. So in this case, it was thyroid tumor. Here you can see on the red, there's cancer area delineated by pathologists in the primary tumor, and in yellow, these are cancer metastasis to lymph nodes. And again, we did unsupervised segmentation similarity. What was important, that inter-tumor similarity, I mean the similarity between tumors from different patients. If you look for the similarity of primary tumors on the one side and the similarity of metastasis on the other side, these are more similar than primary tumors and metastasis from the same patient. So in fact, intratumor heterogeneity when we compare primary tumor and metastasis was higher than interpatient heterogeneity, something unexpected. At the moment, we know this is just the influence of the microenvironment at lymph nodes, which just force some metabolic changes in the lymph nodes. Okay, and final example. What is the major threat in the cancer treatment? Distant metastasis, because majority of patients die because of distant metastasis that develop several years after primary treatment. So we would like to know at the time of primary treatment, what is the prognosis? So in this experiment, we have a cohort of patients with breast cancer. We analyzed the tissue taken during surgery. Then, after 5 years, we've got information whether the patient stayed healthy or developed distant metastasis. And when we analyzed heterogeneity of these patients, we found that heterogeneity was much higher in the patient who remained healthy than in the patient who developed metastasis. It was also pretty surprising. We didn't expect that, but we followed this observation with more refined analysis of the tissue, and we found that higher heterogeneity corresponded to higher infiltration with immune cells. So when we have more immune cells at the time of primary treatment, this is good prognostic marker, because this tissue will respond to the treatment more favorably. Okay, and I told you at the beginning that this is not, at least at the moment, this is not a method for cell biology. What is the problem? The problem is resolution. Okay, so all these images that I showed you were taken with the resolution 50 micrometers. And this is standard resolution we use in these types of experiments, 20 to 50 micrometer, which means that single pixel that we analyze, this is a cluster of few cells. So this is not sufficient for cell biology. Currently at laboratories, people have spectrometers or some infrastructure, I would say, because this is not a spectrometer, that enable a resolution 1 to 2 micrometers, which is enough to say what is the difference between nucleus and cytoplasm. Okay? Maybe from the point of view of microscopy, it doesn't sound seriously, but keep in mind that in one experiment, we can analyze hundreds or even thousands of different molecules. So we have complete picture, molecular picture of our specimen. Again, not yet in the cell biology, but this is a technical problem. So I'm pretty optimistic because technical problems are easiest to solve. So I believe that in maybe next decades, this method will enter also to the field of cell biology. Probably not till next year. So next year during work in the nuclear workshop, I won't use and data received using this method. Okay, at the end, I would like to acknowledge my colleagues from the laboratory of clinical proteomics, and also I'd like to say a few words about my institution and the city. So this is Institute of Oncology in Lviv. So this is a branch of National Research Institute of Oncology. Next year, we'll have 75th anniversary, but the institute as a whole is much older. Next year, we have 19th anniversary because National Institute of Oncology was founded as a radio institute in 1932 by Marie Skłodowska-Curie. And also my city, Gliwice, as you can see, both Gliwice and Lviv were mentioned in the chronics more or less at the same time, at the end of the 13th century. So this is town with some medieval, maybe not architecture, but this medieval urbanic concept. We have Greenock, we have Rathaus, we have castle, and we have the most famous structure from Gliwice, which is the tallest wooden construction in the world. You know it as a Gliwice radio station. It was famous because of the beginning of Second World War. It was so-called Invincible provocation. But anyway, especially at night, it is really very scenic. We call it our own Eiffel Tower. Okay, so thank you very much for attention.
I have a question, maybe comment. That's quite interesting to see the similarity between primary and then those specimens that you analyze from lymph nodes. It is a notion similarity within groups, right? It is a notion in the field that lymph nodes are the endpoints of migrating cells, and that nothing will happen after the cells which will enter circulation. They will be trapped by lymph nodes and they are not going anywhere else. So what might be interesting, and I wonder if you have done this or you plan to do it, is to compare primary lymph nodes and distant metastasis either in liver or in bones or in liver somewhere else where they are really cause of death and suffering of patients.
Well, okay, this is a very complex answer to this question because the situation is very complex. And there are different hypotheses what is the role of this lymph node metastasis. So one of them is what you expressed. There is technical problem, maybe not technical. Well, this is a basic problem with this type of analysis. Because when we have metastatic disease, very rarely metastatic disease is treated with surgery. Okay, great. So this is the problem that we really don't have material from metastatic disease other than biopsy, which is used for molecular
Well,
diagnosis.
it is post-mortem, but
Okay, but this is not the basic treatment. If there is metastatic disease, there is no surgery, so this is the shortage of material for this type of analysis. Yeah. But the concept is very interesting that probably we'll try if we have material to address this type of questions.
So thank you for a very nice talk. I have two questions. First, you use molding, right? Matrix assisted mass spectrometry. So that means you have to mix the sample with a matrix. So I think it would be difficult to identify very small metabolites because you will get a lot of noise coming from the matrix, right? That's one thing. Another thing, with this method, you cannot do the quantitative analysis because then you would have to do in vitro staining, for example, isobaric staining or something like that. So basically, you would just be able to say yes or no, right? But if the difference is quantitative, if you compare the samples, you will not be able to say to see any difference. That's a huge
In fact, you addressed two major weaknesses. I will start from the second one. In fact, there is few laboratories that they work just on the quantification of imaging. There are some concepts, they are not general. I believe that in future we will go into quantitative mass spectrometry imaging. After years, we get into quantitative mass spectrometry finally. So I believe that also quantitative mass spectrometry imaging is possible. If we have more normalization, these are technical problems. Technical problems quickly define what is the problem, we can solve them.
Oh, do you have to do tryptic digest, right?
Well, it depends. If we do proteomics, if we are analyze proteins, we have to do tryptic fragmentation. This is for sure. But if we analyze lipids or metabolites, tryptic digestion is not necessary. Going back to the first question, yes, this is true that for mass spectrometry, there is some limit. So we cannot analyze metabolites that are on the size of matrix. But the matrix is around 100 titles. So we can see bigger metabolites, so this is not a problem for lipids, for amino acids. maybe not for amino acid. But if this is something bigger than this aromatic acids, we can use this approach.
Yeah, good. I will try to be fast. Okay, so we have always seen this method as very promising for the future because theoretically it would replace all the histochemistry and all the synthetic antibodies and so on. So how do you see it for the future? And second one, when we at cellular biology, what is now the spatial discrimination of this method? Because I know that it has been nearly half a micron already, but I don't know how is it now in this sense.
Okay, I will start with the second. As I said, the spatial resolution at the moment, which is used in few laboratories, is around 1 micrometer. And the problem is not only the resolution of the laser, the problem is sensitivity. Because as smaller is the pixel, as fewer ions get to the detector. So this is two sides of the same coin. Okay, but it is in use around 1 micrometer. Talking about diagnostics, well, my DMSI, this is not very fast method. So definitely this is not for intraoperation diagnostics. It would be rather for biomarker discovery than for actual diagnostics, but this is one exception, not mine. But this approach is used for really diagnosis during the surgery. People have this knife. So when we cut out the tissue during the surgery, we immediately generate ions. And there are some approaches that we can analyze these ions that are generated during surgery. And this is really guided by the algorithm. So surgeon could be guided by the algorithm regarding the risk of going into the margin or keeping in the proper area. This is not margin, but still this is mass spectrometry margin. The different method of ionization.
Is right.
Well, it started to be developed like 5-6 years ago, and there are some hospitals, for example, in Great Britain, when they use this approach in the surgery room. Thank you.
I would like to invite next speaker, Paweł Kozak from Institute of Molecular Genetics, Czech Republic, Prague.
Oh. Okay, it's my enormous pleasure to be here and to meet young and new friends. When I was preparing for this talk, I realized that I have been here at the end of 80s. I found a picture from 80s actually from Lviv, and it was my first visit after the very long time, and it was really exciting to see the development of the city yesterday and the beauty again. So I hope to be back next year to have more time to visit. But as some talks were missing Nucleus, so I will replace my talk only in the Nucleus, and I will try to give you a few hints about very new facts that we have seen over the last 6 years when we and a few colleagues of us started to see interesting functions of nuclear lipids, and more specifically these nuclear lipids in modulating DNA transcription. Okay, so when we look at in a living cell at the nucleus, we don't see really much because in contrast to the cytoplasm, nucleus hasn't got membranes inside. So all the events inside the nucleus are guided by intermolecular interactions, mostly non-covalent. And these events actually make various compartments within the nucleus, like nuclear loops, speckles, and so on. So most of the events happen in specific foci, not just in the diffuse manner. How is this achieved? Largely, it is still a big question, and I might provide you at the end one of the possibilities that contributes to this nuclear order. Generally, only protein inductions, protein-brain acid inductions are considered. We are introducing additional player in this scheme, and that's the nuclear lipids. Okay, so just to keep the story short, what has been known for a decade or so, the complete pathways for nuclear phosphoinositide is present in the nucleus. There are 3 ways how to produce phosphoinositides. There are kinases, phosphatases. In my talk, I will, I go into my talk, not to forget, I just would like to introduce my team of the people who contributed to this work, especially including Margarita Sobov, originated from Ukraine, and other from Russia. Okay. So I will concentrate on the phosphoinositides and more specifically on the phosphoinosite 4,5-bisphosphate. You know definitely this from cytoplasm as one of the principal regulators of cellular events on cellular membrane. Is it possible to switch on these slides, please? Okay. Because I go to super resolution microscopy. Okay, that's the maximum I can do from here. Is it better? Yeah, because I go to super resolution microscopy, because we need to see the details. So this is super resolution microscopy, and we see immediately that we can localize PIP2. I will call this just PIP2 for the short. In many areas of the nucleus. And so here I will need the doctor, maybe this one. No. Okay. All right. Because this is really what otherwise you wouldn't see. Okay, so still not. No, okay. So the nucleus is now something like here, okay, and you see many tiny dots. Each of them is about 70 to 100 nanometers in size. Okay, when we look at electron microscopy, we discover that each of these tiny dots can be visualized with PIP2 on its surface. So these are tiny balls up to 100 nanometers. So what are they? It was for the first time that we discovered the structures in the nucleus, and the question immediately was, what are they functioning for? So we did a microscopic molecular experiment. So I will basically use the slides only as a background for giving you the main facts. For visualization of lipid, the only reliable method is quick freezing and freeze structure replica, which only a few labs in the world know. One of the best ones is Professor Fujimoto in Japan. So we'll learn the technique from him. And you see that on the cell membrane, we see a lot of PIP2, the black dots from electron microscopy. And we were able to localize this also in the cell nucleus. And these were reminding those 100 nanometers structures. Besides PIP2, we localize also ceramide and cholesterol on the surface. Then we switch to very sophisticated microscopy that we have in the lab, because this allows us to discriminate various atoms, various elements. And so we did mapping of phosphorus and nitrogen, when phosphorus maps predominantly for nucleic acid and nitrogen for proteins. And we saw that this is the pilot, and you see that inside there is very little phosphorus or nitrogen, and all of it is located on the surface. So, okay, so it was suggesting that these structures are composed mostly of lipids, and with the FIP2 on its surface, the polar heads facing outside. And then we were starting immediately asking what activities of the cell nucleus can be connected with this. And we started with simple experiments looking at the DNA RNA treatment, because that's what people usually do in the nucleus when we don't know what the structures are associated with. So we disrupt the DNA or RNA and then check whether the structures disappear or not. And it became immediately clear that they are resistant to DNAs, but they are very sensitive to RNAs. Okay. And so that directed us in the towards DNA transcription, of course. And we confirmed by super-resolution microscopy that these nuclear foci characterize related nascent, newly formed RNA transcripts. And it was even more apparent by ultramicroscopy. So this is one of the islets, second, 4, and you see that the transcripts are really located on the surface of these lipid islets in the cell. Okay, we then use enzymatic disruption of the islets in very gently permeable cells. So basically, gently permeable cells, we treat them with lipase, phospholipase C. And then when we see that the lipid islets inside of glucose practically disappear, we check for the level of transcription. But as soon as they disappear, the level of transcription goes also to half of the level. So which has suggested that the structural integrity of this nuclear islets is somehow important for activity of nuclear transcription. Okay, so these are more complicated experiments, but we basically confirm that the PIP2 is in complex also with transcription factors, including very important protein myosin 1, and myosins are very difficult binders, of course, of PIP2, on some, that's known from the cytoplasm. And in a set of this, again, a big experiment, we showed that the efficient protein transcription always requires interaction between this nuclear myosin and PIP2. So we had another player which is important for as a partner for PIP2 for the transcription as the nuclear myosin. And we know that this nuclear myosin also resides on the surface of these lipid structures. Okay, so from this, I would like to conclude that these nuclear lipid islets, there are new undescribed, previously undescribed structures of the cell nuclei. The POL2 transcription complexes are attached to them, and disruption of this nuclear lipid islets reduces the levels of transcription of the channel transcription by POL2. And the interaction between the interaction between myosin and PIP2 is required for efficient POL2 transcription. So, what in fact are these PIP2 islets? Are they dependent on transcription, or are they just integral structure inside the nucleus, and the transcription is formed on the surface? We looked at various organisms, and we could find them evolutionary very conserved. We could find them yeast, protozoa, being humans, mice. So they always seem to be present in the cell nucleus. So it looks like an ancient nuclear structure. And when we inhibit transcription using various inhibitors, they still remain in place. Of course, there were no transcripts, but they were like structural entities residing inside of the cell. So we suggest that there are actually structural features, typical structural features of our nuclei, which help to organize transcription. How this could happen? Let me just summarize before I go to the last few slides about how this might be happening. So this is PIP2 molecule. It's only about 4 nanometers, so it's very tiny molecule. This is one of the lipid islets. They sit on the surface. We are still questioning more about molecules inside of the structures. And when we look at the surface of the nuclear lipid islets, we already know that There's the chromatin, the large transcription complex, lamins, myosins, lacto-alpha. And you know that on the surface, besides 4-5 bisphosphate, there's ceramides and cholesterol. So that's the descriptive side of the story. So let's try to do a little bit more of function. So what are we doing right now? So the story is not definitely far from being finished. We are trying to isolate the structures using various PLC purification and interpretation. And you see that we seem to be coming closer. We have already confirmed by microscopy. So we then do a lot of mass spec analysis and conducted mass spec analysis. And we already came to a couple, not to a couple, to quite a few proteins, and most of them are connected with splicing, RNA-2, and transcription regulators. So it looks like we are really heading in the right direction. So what are the common features of these proteins connected to NLI, so to PIP2? Interestingly, about 60% of them contain KR motifs, and more than 70% contain interstitial disorder domains. And this interstitial disorder domains are interesting features of the molecule because they seem to be, they have been confirmed to be involved in so-called phase separation. And the phase separation is a phenomenon which is generally working in biology, and so it doesn't require any exact cooperation between two molecules, but it's mostly based on biophysical properties. And one of the candidates that we already confirmed is the anti-protein, and this is really, in our eyes, a little putative link between PO2 and nuclear myosin on the surface of the animals, because it has got two pH domains which bind with 2, and it has also this low complexity agent, typical phosphate, and it does phase separate. Okay, so we work on this, and I just would like to conclude my talk with this movie, because it does that in addition to specific interactions, we have to think, especially when inside of the nucleus, about phase separation processes and those processes that can moderate activities inside of the nucleus rather than only switching on or off. So thank you very much.
Sorry, I was first. You were the first. You were the first. So, very interesting presentation. Thank you very much, and thank you for bringing us back to the nucleus. So, first question: this PIP2 and nucleus speckles.
Yes, very interesting question. Speckles, they contain a lot of PIP2 as well.
Great.
And we are currently investigating this, but of course the transcription doesn't occur inside of the speckles, but transcription occurs near to the speckles. So we are now checking about the dynamics.
But they do not overlap completely.
No,
They don't.
no, they don't. They are accepted structures.
Great.
And then there's different protein answers.
Now, kind of comments. You make conclusion that those PIP2 structures are important for transcription based on the EZU or quantification analysis. I guess it can be interpreted in different ways. To directly answer the question about correlation with transcription, you probably should run on experiment, because what you have here probably can be interpreted as stability of transcripts,
Yes,
which can be stored in those domains.
Yes.
So do you plan to do this run-on experiments to check?
Absolutely. Absolutely. I have to
Great.
we have many more experiments. So what we are doing now, we are actually trying to understand the mechanism. So we are actually mimicking now the surface of analyze using nanodiamonds and various other structures, and setting transcription assays in vitro so that we could really fiddle around with components.
And finally, in the same direction, just one comment. It is definitely a notion of importance of this liquid phase separation on transcription was a wonderful review by Tom Yan just so a couple of months ago. And now the question is the notion that potentially the cancers and promoters can glued together with the help of those, so and might be really cool to see what's going on.
Absolutely.
Especially if you have a tool where you can inducibly deplete PIP2 and see how.
Not yet. That doesn't work.
So that's
why we asked you to in vitro assays because it's extremely difficult because there are 3 pathways producing PIP2 inside of the nucleus. Which reside inside of the nucleus, okay? There's one knockout mice, which knocks out only one of the pathways. It's nearly impossible.
Can you block nuclear translocation?
In fact, if we block nuclear cytoplasmic translocation, it doesn't really deplete PIP2 because PIP2 is produced inside as well. So you would need to knock out 3 pathways. Plus block the nuclear cytoplasm and then it's getting too messy.
I have a stupid question. Are all genes equally sensitive to PIP2 depletion? Did you do kind of just
Yes. I cannot answer it at the moment. It's a very important question, but what we suspect that various genes might have various requirements to be associated with PIP2s because we also see already from the presence of some specific proteins that not all NRIs are equal. That there are some factors. But we still don't know why.
Because you can infer many things from this simple experiment.
I just want to also bring your attention to the fact that analyzing rich sequences, which are unstructured and are important for phase separation, they are subject to multiple post-translational modifications, which I showed 1 hour before. So the question is whether you are planning to follow specific modifications, whether they affect phase separation.
Yes, we are, but we really need to dissect certain area because it's getting too wide, basically. So I think we will start with the proteins that seem to be involved in this, and we start with just with a few proteins. But absolutely, we are also going to look
Well, for example, P300 should be there.
Yes, should be there. I think it's there. And we will build in modification of the IDR domain because the IDR domains they are important for phase separation, but it also seems that in some proteins, once they bound to something, that the IDR gets more conformational step up.
Yes.
So it really needs to be looked in some dynamics.
Because they are, dynamic.
Exactly.
They come on and off.
Exactly. So
Thank you very
thank
much.
you very much.
I still have. First of all, have you observed these islets in living cells?
In living cells?
Yes. And have you followed their movements?
Yeah. We tried very hard, but to this moment it's impossible. Because PIP2 is very small molecule. As soon as we add anything fluorescent to it, it dramatically changes its properties, and we couldn't do that.
Okay, and what about specific blockers of phosphatases and kinases? Because you suggested that PAP2 is produced inside the nucleus, and in this case there are very conventional tools to manipulate production of PAP2, and it would be quite interesting for me to check if this blockers of phosphatases or kinases change transcription of the nucleus.
Yes, we discussed with few labs who deal with it inside cytoplasm, but decided for this moment that we will concentrate on the specific partners because otherwise we are affecting we cannot, we have no specific inhibitors for the nucleus. You know, and then.
Yes,
that's
these
why
are specific inhibitors for, okay, they can penetrate in the nucleus and work as
Yes.
as blockers. It's
But,
not
but at the same
all-
time, you block the cytoplasm. And the problem is that especially the cytoskeleton is extremely connected to the nucleus. And as soon as you start manipulating the cytoskeleton and actin, especially actin, you get severe effects also in the nucleus. So we are a little bit.
But you can use different kinds of cell lines. For example, KRAS mutant versus wild type, because they have a major differences.
Yes, there are probably ways, but we haven't considered.
And the last question, which is quite interesting for me, there are a lot of PAP2 in the cytosol. And have you observed these islets in cytosol, or they are specific only from? You have a lot of EM.
But this is difficult to answer because in cytoplasm you get a lot of labeling. There's one thing which I didn't mention. These nuclear islets, they haven't got typical membrane on the surface. They are not micelle. So I think that there's a big difference from the cytoplasm where you can typically find this.
It means that it depends on specific interaction with proteins, which are
Looks like which are stabilizing.
stabilizing them only.
Yeah, because otherwise, why are they all of this unified sizes? I think there might be some biophysics going behind it, some entropy stuff that keeps them between 70 and 100 nanometers. Why don't they fuse from?
Thank you.
На 10 хвилин перерви. Прошу повертатися в зал 12:10, ми знову починаємо, продовжуємо нашу сесію. 10 minutes break.
So the next speaker is Olivier from the University of Burgundy in France. And before he starts his very interesting talk, I would like to invite young people in the audience to ask questions, because we are just asking questions between ourselves, and maybe there's something that interests you, maybe something you do not understand. Please do not hesitate. It's normal to ask questions, and this answering questions and asking questions is probably the most interesting part of the talks of the discussion. Alex.
Thank you. I'm very happy to be here, and thank you to the local organizing committee of the 27 on cell nucleus. We are slowly because of COVID pandemic, but moving from one city, beautiful city, Dijon, to another beautiful city, and one of the distinctive features of this workshop is very rich social program and the possibilities to communicate between young researchers and senior researchers, and we started yesterday. It was a very good beginning, only with the senior researchers for now, but I hope that next year we will have full success and continue the tradition of the Kenyan Math workshop. Now, today I will talk about the neutrality of neutrophils. So it's kind of board game and my neutrophils. First, if you remember your blood analysis, blood formula, you will find that among leukocytes, the most represented cells, white blood cells in the blood, are neutrophils, right? And they have several very important functions in the immune system, like production, mitosis, and maybe some functions are missing here. So phagocytosis was opened, everybody, by a scientist who was born in Ukraine and who was working in Ukraine and teaching in Ukraine, and then, he moved first to Sicily to make some studies, and then to France, where he got Nobel Prize for discovering of phagocytosis. Please remember that he was not working initially with human subject. He was working with the sea stars, right? And he first saw and this phagocytosis event in sea stars, and only after that he interpreted, and this discovery was transferred to the human blood cells and the neutrophils with macrophages and some other myeloid cells are phagocytes. So remember that, and please also remember to respect science as a future physicians, because frequently, especially in Europe, in the United States, maybe it will be different, but in Europe, the attitude and the relation between researchers and physicians are quite competitive. And this is what my friends from medical schools, when I asked them, what do you think about science and research, that's what they was the answer. And today we actually will talk about how to make such experiments as he described with mice. Mitosis. Mitosis is another feature of neutrophil, and originally it was described, and evolutionarily probably the aim of mitosis to help the body to trap and inactivate pathogens. As you see here, some bacteria were trapped in the nets. The nets formed by chromatin, specific program type of cell death that happen in response of many factors, in activation in pathogens and activation of TLF innate receptors. So very interesting and beautiful feature to study, and also very important because I told you about COVID first lecture, and neutrophils and mitosis involved in COVID pathogenesis and because of immunothrombosis. You probably know, yes, that one of the complication of COVID is different types of probability, and frequently patients die from that. And mitosis, sorry, and mitosis actually responsible for assembling this electron with reacting with activated by COVID infected cells, platelets, and also responsible for cytokines shown frequently observed in COVID patients. But the real example of neutrality of neutrophils is tumor microenvironment. Of course, we don't have time today to talk about all these cells and their roles in regulating tumorigenesis. And this is very hot thematics, and you know probably that our professors were teaching us about wild effect that now is considered not so right. These mechanistic switches of cancer cells actually not happening in cancer cells, it's happening in tumor microenvironment, particularly in cancer-associated fibroblasts. To simplify, we will discuss all the relations between myeloid cells and lymphocytes. So lymphocytes are main effector cells in our body, our immune system, that are responsible for killing, right, for eliminating tumor cells from the body. And main actors here are NK cells and CD8 killer cells. So neutrophils and also macrophages, another type of myeloid cells, they have from the beginning, they're showing some positive features and helping immune system to fight cancer, but also they could be distracted by cancer cells to become very bad cells and help cancer cells to protect, to shield themselves from immune system from lymphocytes by inhibiting lymphocytes functions. So we recently published a paper about how to reprogram neutrophils so that they could become positive cells and start helping us to fight cancer. And we will talk about this reprogrammation. So we're using different approaches, and the main approach is reverse genetics. So we're constructing a lot of mice models, and with the aim to not just delete the gene in the whole body, but delete the gene in specific cells. Because it's very important. You saw how complex tumor microenvironment is. So that's why it's very important to make a specific deletion in a specific type of cells. And for that, you know, the system is scientists using Cre recombinase, and Cre recombinase is an enzyme that recognizing very specific 36, as I remember, nucleotides site in the DNA that you insert into genome. This is called LOXD size, and it could very specifically excise to cut off the gene out of genome, making compromising the function of this gene. So very specific action, probably there is no such sequence in our genome, so it's only excising this gene, the target gene of interest. And we deleted the phosphatase. Nick introduced this phosphatase as a negative regulator of p53, very important gene suppressor. This phosphatase called WIP1, wild type use phosphatase, or genetic name is EPM1B. Protein phosphatase, manganese dependent 1 delta. So we deleted this phosphatase in hematopoietic stem cells, and you understand that in hematopoietic stem cells in bone marrow, so we will get all hematopoietic lineage compromised, and phosphatase will be not active in all hematopoietic cells, as shown here. Epithelial cells, here we use fluorescent protein like an antibody, and we stay with this reporter, and we see that it stains cells only inside intestinal villi, where blood cells are located, and not staining, not staining anything in intestinal epithelial cells. But the systematic of today's talk with myeloid cells and neutrophils, we also use several models to delete this phosphatase in myeloid cells, in neutrophils mainly, and partially in monocyte macrophages. So this is at the bottom where we see one model delete around 70% of neutrophils will be affected, and in the second model, the 8-3 conditional model, we have about 80% of neutrophils affected. So basically, what we found that if you have overexpression, so if you have a lot of phosphatase inside the cells, tumor will grow much faster than this green curve, and then in wild type, in the black curve. But if you delete WIP1 in hematopoietic cells or in myeloid cells, you significantly delay the tumor progression, and tumor will not grow. And you can even treat with this WIP1D deficient neutrophils mice, and you injecting and transplanting neutrophils to the mouse will have mutilate progression. Immediately when we stop the treatment, the tumor starts to grow faster. And this is of course showing the importance of neutrophils in regulation of tumor growth. But I think the most important finding that we have is that action of neutrophils is going through regulating environment, called reprogramming. It's reprogramming the transcriptional program of neutrophils. And now, when we delete P1D, because it's negative regulated P53, we activate P53, P53 transcriptional factor, among targets that Nick showed to you. There are also immune targets, including activating lymphocyte activating lipids that now expressed on the surface of neutrophils, and they activate killer cells, and that's how our immune system start more become more aggressive against the tumor cells. So actually we can add this ligand presenting feature of neutrophils on the surface as another property of neutrophil cells. And look why we're talking today, we're supposed to talk about nucleus, about neutrophil, because we, in all this function, we study very interesting nucleus that neutrophil has this segmented type of nucleus with such rich functions and synthesis function, and now we initiating the project on synthesis with Alexei to study the role of nuclear membrane, nuclear envelope in mitosis. So to simplify, so we understand that we had neutrophils and lymphocytes that are important in fighting cancer, and tumor cells could suppress both lymphocytes and neutrophils. Neutrophils are in thinking mode what to do. Usually in this instruction from tumor cells force neutrophils to become inhibiting cells and inhibit immune response. So these features that we found also could be used in developing new strategies of immunotherapy. We are working with the former postdoc of NIC, Alexei Petухov. He's developing CAR-T strategies, and we know that CAR-T is very promising anti-cancer immunotherapy that actively used in the United States, but unfortunately in Europe it's not allowed in most of the countries because of the law. And now it's only research model, or maybe some clinical studies only. Basically, what is CAR-T? It's changing, introducing a new genetically engineered receptor with when you change the recognizing part of T cell receptor for the antibody that's specific to cancer antigen or antigen that express on cancer cell. The main one now is B cell marker CD19. Then they have adapter region, transmembrane region, signaling region, and also activating region with where they use the same ligand that we found very important on the surface of neutrophils to activate inside to potentiate the signaling inside the lymphocyte. So the strategy worked like physician taking blood from patient, separate lymphocytes and infect lymphocytes with lentiviruses that bearing this chimeric antigen receptor, and then select the cells and inject back to the patient, and this efficiently kill all very specifically kill all tumor cells that labeled with antigen matrix. So in this situation, right, what we are now trying to do to show that neutrophils, so here is a modified lymphocyte that is much enforced against the tumor cells, and then we by modifying and reprogramming neutrophils, we potentiating the anti-cancer immunotherapy. I think I will stop here, and if you have questions.
Thank you very much. I have a few questions. Number one, so a good friend of mine, you know him very well, Hans-Uwe Simon, who worked on mitosis. They have shown that the dysfunction of mitochondria severely affects the whole process of mitosis. Do you have any thoughts or any information on whether the viral proteins may affect mitochondria? Or, for example, week 1 can affect mitochondria?
With one as P53 target and as
Well, independent of
regulator, I don't have information that I can could confirm this data that independently of P53 with one could affect directly the mitochondrial function. And when we're talking about mitochondrial function, we didn't discuss today mitosis in details in the molecular mechanism, but the main trigger of mitosis is active production of reactive oxygen species inside the cells. And as you know, right, mitochondria could be alternative. Of course, in neutrophils, the specific system, the neutrophils is ROS producing cells, and they're using this ROS to reactive oxygen species to kill pathogens. Okay, so there are a lot of systems that damaged or not damaged, like mitosis program of cell death. Using to amplify the reactive oxygen signaling to initiate mitosis. And mitochondria, you know, it's an alternative source of reactive oxygen species that produced in all the changes in potential. And because I was just doing experiments, and what we found just recently, last month, with this using this sophisticated seahorse machine that you know very well, that there is changes in mitochondrial breathing. Okay, changes. Yes, OX4 in changing in OX4. Actually, myeloid cells are more glycolysis dependent, yes, but glycolysis dependent, and lymphocytes are more oxalate dependent. The question was not yes or no. The question, do you know something? I know something. Yes. You can talk very long.
Okay, I'm not supposed to ask you any more questions.
Yes.
Roman, I mean, probably no need to present the next speaker, but still, Roman Besic from the National Medical University is going to talk to us about science in Lviv.
Yes, science in Lviv, science in medicinal chemistry and pharmaceutical chemistry. And my talk will be in Ukrainian. Я хотів би сьогодні у своїй короткій доповіді презентувати в історичному ракурсі розвиток фармацевтичної і медичної хімії у Львові, яка завжди була пов'язана із кафедрою фармацевтично-органічної і біологічної хімії нашого університету. І так ретроспективно хотів би представити, власне, сьогоднішньому товариству основні досягнення і як розвивалася, як змінювалася фармацевтична хімія у Львові. Почав би з того, що в даний час відбулися революційні зміни у самому процесі створення лікарських засобів, і коли був, як називається, романтичний період фармацевтичної хімії, коли маленька група могла зробити свій лікарський засіб. Це, наприклад, була група Павля Егліха із його знаменитим Сиборсаном, який почав еру хіміотерапії, чи, скажімо, Олександра Флемінга, яка відкрила антибіотики пеніцилінового ряду. В той час, звичайно, були великі проблеми, але це був романтичний період, тому що невелика група вчених могла дати світу новий лікарський засіб. Були великі проблеми, тому що могли синтезувати багато сполук і вивчати, але обмеження були дуже великі до біологічної активності, до вивчення біологічної активності сполук. Зараз ми маємо зовсім іншу еру, коли вступила комбінаторна хімія, віртуальні скринінці, хемометрія, PCR-аналіз, молекулярне моделювання і особливо високоактивний скринінг. Зараз ми можемо тестувати одну молекулу на велику кількість активності, ми можемо тестувати тисячі молекул на різні види активності, і це, звичайно, привело до змін у створенні лікарських засобів. В той же час цей процес став дуже кошторисним, і відповідно зараз малі групи вже не можуть бути повноцінними гравцями на ринку створення лікарських засобів. Необхідно великі колективи. Підраховуємо, що цей колектив може бути до 150 різних вчених, і бачите, така класична схема створення лікарського засобу, вона триває від 10 до 15 років, і що головне, ця інвестиція повинна бути до 2 мільярдів доларів. Відповідно, це зараз прерогатива великих компаній. Тим не менше, я вважаю, що малі групи можуть знайти своє місце у пошуку лікарських засобів. І, власне, я хотів би це так екстраполювати на дослідження, які проводяться на кафедрі фармацевтичної хімії. Ми маємо дуже довгу історію, я не буду в ній зупинятися. Перше, це 1853 рік, в якому була заснована наша кафедра фармацевтичної хімії. Вона була пов'язана з будинком старого університету в 1854 році, в якому і виник, до речі, фармацевтичний факультет. З 1930 року кафедра перейшла до колегіум фармацевтикум, це старе фото, будинок, який був побудований за кошти Галицького фармацевтичного товариства, яке виділило шалену суму на той час, це було 500 тисяч злотих. І, власне, побудувало корпус фармацевтичного факультету для того, щоб навчати своїх студентів у Львові і окремо від університету. Історія нашої кафедри була зв'язана з видатними постатями. Перший це був Франц Лес, який організував кафедру нашу фізичну. Він був дуже коротко, тому що він проводив дослідження на тодішньому сучасному європейському світовому рівні. Він відкрив властивості озону, деяких органічних сполук, відкрив пеларгонову кислоту, і досліджуючи цю кислоту, він втратив зір. Другою видатною постаттю був Едвард Лінеман, професор і завідувач кафедри, він вивчав аліфатичні склади. І перше, це було досить прорив органічній хімії, він відкрив перетворення вуглецевого шпілету. Я хотів би звернути вашу увагу, що це була політика Австро-Угорщини в цей період, відправляти на периферію відомих вчених. Вони повинні були працювати відпрацювати певний період у периферійних університетах, яким все-таки був на той період Львів. І тому у нас була така видатна особистість, як Леопольд Певель. До речі, в його рідному місті Грації є вулиця Певельштрасе, а саму історію австрійської хімії називають ерою Пебаля в той період, коли він працював. Ну, його одним із найбільших відкриттів це, власне, синтез кетонів, які цинку на хлорангідриди. Він цю реакцію відкрив разом з Августом Фреудом, ще одним видатним представником органічної хімії. І, власне, ця реакція була одною з визначальних у елементах органічної хімії. Тим не менше, це була доволі цікава і яскрава особистість. Можливо, та яскравість закривала до того, що він загинув досить трагічно. Під час експерименту він подискутував зі своїм аспірантом, щось не поділили, його аспірант вбив. Завжди кажуть, з аспірантами треба проводитися чесно, що старався робити в своїй діяльності. Друга особистість – це Олександр Фреуд. Він відкрив циклопропан, який використовувався довгий час при наркозі. Окрім того, він увів свою видатну роль, яку чомусь часом недооцінюють в історії польської науки. Він був першим автором польського підручника з органічної хімії польською мовою. Броніслав Радзішевський, ректор університету, крім того, завідувачем кафедрою, видатний вчений, ввів дві імунні реакції. Перша – це реакція умилення нітрилів, і друга реакція синтезу імідозолів. Я завжди думаю, що це, напевно, прообраз нашого наукового напрямку, тому що це перше були гетероцикли синтезовані. І ще один видатний вчений, Богуслав Бобранський, який іммігрував до Вроцлава, і там став ректором університету. Він проводив системні дослідження гетероциклічних сполук, ввів у медичну практику такий засіб возальгін, це похідне барбітурової кислоти. Він використовується досить час, але використовується з такою не зовсім доброю метою для виконання смертної кари у деяких штатах Сполучених Штатів Америки. Тим не менше, автором цього препарату був Владислав Богранський. Власне, це він вже в період, коли був ректором Вроцлавського університету. Я, до речі, був завжди ці показую слайд, тому що чоловік дуже добре володів українською мовою, і це видно по його записах і по його біографії, які вони йшли в архівах. Професор Микола Товкевич на посади особисті вже радянського періоду, він вів 4 препарати в медичну практику, став автором першого підручника українською мовою і, власне, є основником нашого теперішнього наукового напрямку, це циклічних сполук, сіркоазотомісних. Тут на цьому слайді представлені видатні особистості, видатні вчені, які працювали у нас на кафедрі, які розробили ключові методи синтезу похідних біолин-трикіазану, тіазолітину, різних гетероциклічних сполук, потенційних лікарських засобів. Ну, я хотів відзначити професора Йосипа Дмитровича Комарицю, який вперше запропонував реакцію Ретро-Гіпса-Альдера, яка, я думаю, що потенціал цієї реакції, створений на основі нових молекул, ще буде оцінено в майбутньому. Професор Музиченко Володимир Панасович, який ще працює у нас на кафедрі, він ввів методи електрохімії, утворення важких сполук. Теперішній ректор нашого університету, професор Семенковський Борис Семенович, він, власне, розробив нові методи створення сполук дуже складної структури з використанням надзвичайно простих хімічних перетворень, і вперше він був ініціатором введення таких інсіліко методів до дизайну молекул, до дизайну складних структур, до дизайну самих ліпоподібних молекул. І, власне, на цьому і розвивається зараз тематика нашої кафедри. Працюємо в галузі тіазолідонів, дуже цікавий напрямок, досліджень, немає часу на ньому зупинятися, але хотів би сказати, що вже навіть в цій групі є введені лікарські засоби, в тому числі препарати-агоністи, похідні тіазолідіони, інгібітори дозоредуктази, діуретики, різні антимікробні засоби і ці протизапальні лікарські засоби. Ну, в сучасному стані ми працюємо, власне, в цих сполуках, і в цьому класі сполук дуже це ж були присутні аспіранти, які долучилися до синтезу. Ми маємо достатньо довгу бібліотеку різноманітних гетероциклічних описників сполук, які тестуються, які мають протиракову, протитрапаносомну, протитуберкульозну і противірусну активність. Це основні вектори дослідження, які проводяться на нашій кафедрі. Одним із найбільш успішних це є пошук молекул, які мають протипухлинну дію. Тут ми маємо досить хорошу співпрацю з Національним інститутом раку в Сполучених Штатах Америки, де проводиться тестування сполук методом високоефективного скринінгу, і серед 7 тисяч молекул, які ми вже встигли вивчати, зараз маємо до 10 сполук, які проходять поглиблені дослідження, реакції представлені на цій схемі. Маємо дуже хороший набір хімічних реакцій, які ми використовуємо в своїх дослідженнях. Це і методи мікропольового синтезу, і методи зеленої хімії, особлива гордість за моїх учнів, які розробили полікомпонентні реакції, тандемні, доміно процеси. Це дуже важливо, вони проходять віреліво і стереоселективно. Відповідно, це дозволяє дістати молекули. Дуже так швидко хотів би показати. Практикуємо в своєму дослідженні так званий гібрид фармакопорний підхід. Власне, от на цьому прикладі він дуже цікаво представлений. Синтезуємо напівсинтетичні молекули, які мають природну матрицю у своїй структурі. Ну, от, наприклад, із омили білої, віском альбум, одержимо тетрапеновий глікозид, який модифіковано в реакціях хімічних, додано гетероциклічне ядро, і результат ми дістали молекулу, яка має досить сильну, потужну селективну протилихімічну дію. Наступний слайд, будь ласка. Тут на цьому слайді є представлені методи, які ми використовуємо в математичному моделюванні. Зокрема, останнім таким досягненням було те, що ми розробили фармакофорну модель протипухлинного потенційного засобу, синтезували на його основі молекули і тестували, виявили досить спортивні сполуки. Ряд реакцій теж представлено власне на основі цієї моделі. Ми одержали таку досить цікаву потужну молекулу, яка проявила високу селективну протипухлинну дію. Не буду зупинятися на цьому. Дуже використовуємо у своїй роботі міжнародну співпрацю. Тішуся, що ми маємо дуже велику таку, скажімо, мережу колег з інших країн, які задіяні в наші дослідження. Зокрема, один із них це є дослідження протипаразитарної дії. Ми встановили, що наші сполуки мають досить високу протизапарну дію, і це продемонстровано в кількістю публікацій з цієї тематики. Співпрацюємо з професором Філіпом Брельє із Парижу, з музею історії природи. Також, незважаючи на те, що основна протипухлинна тематика, ми знайшли ряд сполук, які можуть бути потенційно цікавими для терапії вегеневого фіброзу, протифіброзна дія. Насправді арсенал сполук є дуже обмежений для такої активності, і це ми проводимо у співпраці з професором Майком Бастом з Маастріхту. Ну і, звичайно, механізм протипухлинної дії, вивчення наших сполук, тому що я вже казав, це дуже велика колекція цих молекул, які мають селективну протипухлинну дію. В цьому напрямку ми маємо певні успіхи. Мені приємно, що ми співпрацюємо з професором Єгором Басецьким, так само з професором Ростиславом Стойко, який сьогодні був на конференції, також з професором Яном Мінським з університету Зополя, ну і з професором Робертом Прокраловичем з Центру молекулярної медицини і Академії наук Відня. Виконуємо політику, яку нам старається впроваджувати наша міська рада. Ми стараємося максимально працювати із різними країнами. І я нарахував сьогодні день прапора, тому я зробив такий слайд. 20 країн співпрацюють у нашому проєкті, у проєкті фармацевтичної хімії. Власне, і насамкінець хотів подякувати тим, які створюють це нове обличчя фармацевтичної хімії Львова, це молодим людям, які мають великий потенціал, як на мене, які мають бажання, які хочуть працювати, які люблять свою професію. І я надіюся, що вони рано чи пізно досягнуть прогресу у створенні подібної молекули. Дякую.
I have a question. So, are you doing? Yes. No. Are you doing HTS only, or you can do also high content screening, which is cell-based?
Not only ITS. On the first stage of testing of our compounds, we use ATS to receive some type of structure activity relationship. After we select, we select some key compounds and use another type of screening.
Thank you.
Thank you.
So the next speaker is Anna from the Lviv Institute of Technology. It's my pleasure to introduce her. And as we are in the native city of Leopold von Sacher-Masoch, she's going to talk about pain.
Thank you. Thank you, the organizers, for inviting me here. And yes, I will try to do my talk in Ukrainian. Ну, я хочу всіх поздоровитися, привітати з нашими святами, з Днем Прапора, з Днем Незалежності, побажати всім здоров'я, успіхів та мирного неба. І хочу нагадати, що вчора був ще одне свято, моє професійне свято як нейрофізіолога. Вчора ми святкували Міжнародний день мозку. І я хочу нагадати, що мозок людини – це найскладніша організація матерії у Всесвіту. І тому від того, як кожен з нас управляє, керує цим органом, залежить і добробут нашої країни, і її незалежність. Тому я дуже часто читаю популярні лекції про те, як розвивається наш мозок, як підвищувати його потенціал. І сьогодні я теж підготувала дуже популярну лекцію, вона буде не зовсім наукова, але дуже популярна, про таке прикладне питання нашого, яким теж керує наш мозок, про біль. Ну, як будь-яка нормальна людина, я особливо в дитинстві, тому що я дуже боюся болю, я дуже часто задавалася питанням, а чому ось ці вчені, які крутять хвости мишам, як нам казали попередні доповідачі, чому вони не позбавлять людство від цієї напасті, від от такого неприємного відчуття болю? Ну, давайте розбиратися. Біль, насамперед, це фізіологічна функція організму, яка захищає нас від будь-яких поразників зовні та всередині організму. Тобто вона попереджає нас від того, що може зруйнувати наш організм. І завдяки цій функції ми можемо реагувати на ці подразники, так висмикнути руку від гарячого або гострого і вилікувати, наприклад, зуб. На жаль, існують люди, які не відчувають болю. Це дуже рідкісне захворювання, яке робить людей зовсім нечутливими до болю. На жаль, такі люди дуже рідко доживають до дорослого віку, тому що вони можуть просто померти від зараження крові, від того, що від запалення апендиксу, від серцевого нападу. І, на щастя, таких пацієнтів дуже мало, там біля 100 людей на нашій планеті страждають від цього захворювання. Зараз з цим нема чого робити, це тільки можна відреагувати, вилікувати лідеруванням геному, що зараз неможливо. І інша сторона цього питання – це нестерпний хронічний. Це коли ця нормальна захисна функція організму перетворюється на вже некорисну функцію і просто псує життя людини. І часто такий нестерпний боль стає причиною суїцидів або евтаназії, якщо це дозволено в країні. І від такого хронічного болю страждають біля 10% людей на планеті. І з цим є чого робити. І багато лабораторій намагаються вирішити цю проблему такого вже нефізіологічного хронічного болю. Зараз існує новий підход, нова концепція розглядати хронічний біль як окрему клінічну сутність. Тобто не відокремо від основного захворювання, яке визвало цей хронічний біль. По-перше, це із-за того, що не завжди ця хвороба може бути вилікована. А по-друге, тому що іноді хронічний біль існує навіть тоді, коли вже не існує цього захворювання, яке його визвало, завдяки тому, що молекулярні механізми, які вже перебудувались і працюють у цьому патологічному стані. І тому найголовніше завдання вчених – це фенотипування болю, тобто знаходження пов'язання конкретних молекулярних механізмів з тим чи іншим типом болю. І ось цим займається і наша лабораторія. Ну, давайте розберемося, як працюють системи нашого організму, що задіяне в генерації болю. Все наше тіло пронизано відростками нервових клітин, які можуть реагувати, збуджуватись у відповідь на зовнішні подразники. І потім цей сигнал потрапляє в головний мозок, де і формується емоція болю. І від того, верніше, якої інтенсивності і тривалості буде ця емоція, залежить від того, як цей сигнал первинний був відпрацьований в містах переключення. І одне із найважливіших місць – це дорзальний рог спинного мозку, де, собственно, і відбувається обробка цього сигналу. І давайте зараз подивимося, як сигнали поширюються дротами нервових клітин. От як бачите, цей процес нагадує протікання струму. Ну, власне, це і є електричний струм, які переносяться зарядженими частинками, іонами. І при перенесенні електричного заряду від клітини до клітини, ось так заряджені частинки перетинають мембрану, а потім в містах, де є переход сигналу від одної клітини до іншої, це називається синапсом, ось завдяки нейромедіаторам відбувається перенос електричного сигналу до іншої клітини. І таким чином сигнали, в тому числі і больові, передаються з периферії в наш головний комп'ютер, головний мозок. Серед іонів, які беруть участь у передачі сигналів, іони калію, натрію і кальцію. І саме іон кальцію відіграє дуже важливу роль. Його називають універсальним посередником, тому що саме зміна внутрішньоклітинної концентрації іонів кальцію і регулюють всі основні фізіологічні процеси від експресії генів і до загибелі клітин. І давайте подивимося на схему. Ось. В будь-якої клітини концентрація іонів кальцію в внутрішній клітини, в цитозолі, дуже низька, порядка 100 наномолів. А ззовні дуже висока, порядка 2 мілімолів. Тобто різниця у 4 порядка. І кальцій після періоду електричної активності, після збудження, може потрапити в клітину ззовні усередину через спеціальні канали або рецептори, вивільняться із внутрішньоклітинних органель. Але це має відбуватися дуже швидко. Потім вмикаються системи, які відновляють концентрацію кальцію. Тобто це насоси, помпи, які виводять кальцій у зовнішнє середовище або назад в органель. І от на цей процес дуже енергозатратний. І на це може в деяких моментах витрачатися до 90% енергії організму. Тобто це дуже важливо для кожної клітини, щоб підтримувати цю дуже низьку концентрацію кальція всередині клітини. І для чого це важливо? Для того, щоб регулювати всі процеси. Я вам покажу декілька приладів. Якщо можна, звук, вимкнути звук комп'ютера. Вимкнути? Так, ось цей фільм, він зроблений студентами Американського університету. Ви бачите яйцеклітину, яка в момент запліднення сперматозоїду. Він проникає в клітину і бачите, як концентрація кальцію, яка змінюється від блакитного, низького до червоного, підвищуються клітини. А це 3D-конструкція, бачите, у цьому місці проникнення сперматозоїду, підвищується кальцій, і після його підвищення в цитозолі виникає друга хвиля внутрі ядра. І саме ця хвиля підвищеного кальція внутрі ядра, бачите, Nucleus movement, і генерують початок діління, тобто початок нового життя будь-якої клітини. Бачите, цей пік усередині, це підвищення кальцію всередині ядра. І ще один приклад, бачите, тут зафіксована лічинка рибки Даніо. Генетично кожен нейрон там загружений кальцієвим барвником, який реагує на будь-які процеси. І от слідкуйте за цим, зараз ви бачили цей спалах. Це в той момент, коли їжа просто проплила повз цієї рибки. Отак її нервова система відреагувала на те, що дуже важливе для цієї рибки, тобто реакція на їжу. І ще один приклад, бачите, це вже нейрони в культурі, нейрони переднього мозку, вони теж, в них теж є кальцієвий барвник, він змінюється. Можна ще раз запустити цей новик. Тобто, коли клітини як-то фізіологічно активні, вони змінюють концентрацію кальцію. Але можна ще раз там? Ні. Ось тут має бути. Ви бачили, що всі клітини змінюють флуоресценцію, крім ось цього нейрону, який завжди був стабільний у своїй рівні флуоресценції. Тобто ця клітина вже померла, тому що вона перевантажилась кальцієм, системи відкачки не спрацювали і клітина загинула. Тобто зміни концентрації кальція дуже важливі від початку життя клітини до її загибелі. Повернемося до болю. Я хочу вам показати такі рецептори, які існують в синапсах майже всіх нервових клітин. Вони реагують на глютамат, це основний нейротрансмітер в головному мозку. І вони ці рецептори називаються ампа-рецепторами, вони такі дуже рухливі. І вони приймають активну участь в багатьох когнитивних процесах, в навчанні, в запам'ятовуванні, в мисленні. А в спинному мозку вони приймають участь в передачі сенсорної інформації, в тому числі передачі больових сигналів. І ці рецептори в нормі вони не пропускають іони кальцію. Але якщо є патологічний стан, наприклад, біль, вони починають пропускати кальцій. І от що відбувається. Якщо в нас є якесь там джерело болю, то кальцій потрапляє в клітину через інші рецептори, але цей кальцій може активувати протеїн кіназу С-альфа, цей білочок відповідальний за зв'язування того нормального ампорецептору з молекулярним якорем, який утримує цей рецептор у мембрані. Після того, як ця зв'язь порушена, на місто того нормального рецептору завдяки її такої рухливості амборецептора приходить цей патологічний рецептор, який може пропускати кальцій. І дуже багато кальцію входить в клітину, і це змінює просто всю сигналізацію цієї клітини, роблять її більш збудливою, і сигнал вже передається не той, що в нормі, а ті, що відповідають больовому такому стану. Що можна зробити в цій ситуації? Можна просто заблокувати рецептор, як це робиться за допомогою звичайних фармакологічних агентів. Тоді ми блокуємо всі процеси, в тому числі нормальні, а я казала, амборецептори задіяні і в проведенні просто іншої сенсорної інформації, не тільки больової. Тому ми вирішили піти іншим шляхом. Ми збудували таку коротку молекулу ДНК, яка комплементарна матричної РНК, яка транслює саме цей білок протеїн кінази С альфа. І якщо таргетна цю молекулу ввести в ту частину організму, в ту частину спинного мозку, де відбувається перетворення цього больового сигналу при хронічному болю, то ми блокуємо цей процес, і ампорецептори працюють, як вони мають працювати в нормі, не перетворюючись на таку патологічну кальцієву форму. І ось цей метод реально дозволив нам у наших лабораторних тварин дійсно позбавити їх відчуття болю, при тому, що джерело цієї болі, у нас було периферичне запалення кінцівки, воно нікуди не дівалося, а відчуття болю, воно зникало. І цей собственно, процес, коли ми через катетер вводили цю молекулу, ми називаємо їх антисенсові олігонуклеотиди, вводили до люмбарного відділу, де у нас перетворювався цей сигнал від запаленої кінцівки. І таким чином ми саме в цьому відділі спинного мозку блокували транскрипцію оцього білка таргетного, який і був відповідальний за перетворювання нормального рецептора в патологічний. І я маю зазначити, що така технологія застосування, вона вже, це новий підход, але вона може бути аплікована на будь-які захворювання і на лікування будь-яких захворювань. І в Сполучених Штатах Америки вже є препарат Вітравен, який отримав дозвіл FDA, за допомогою таких антисенсорних нуклеотидів він лікує цитомегаловірусну інфекцію сітківки. І ми вважаємо, що, в общем-то, це такий такий перспективний підхід, який може не тільки зупинити біль, але ж і лікувати і багато інших хвороб. Дякую за увагу.
Questions?
Well, actually, I have a question. So, in this approach, for how long after injection, animals are protected against acute pain?
In our case, it was 4 days.
4 days.
4 days. Yes, it is quite a long period for such a
And
severe pain.
Because I'm sure if you consider application in the direction of chronic pain, which can be caused by different type of pathologies, including some terminal stages of cancer, where unfortunately we have to sedate or do some of the painkillers, which have some tremendous side effects for patients. Do you think that this approach can be used also in cancer patients?
Yes, we think, especially because of absence of side effects, it would be very promising. And we are thinking about to somehow to insert some pump, probably, which will be continuously inject in locally these molecules and produce.
My understanding is, again, probably I'm wrong, that it is an approach to permanently damage with some inhibitors those receptors in patients using some of the cacti analogs, but over there damage would be permanent, and here you have this window of opportunity to repress pain for several days, and then in case the patient would recover, he or she would have a normal life.
Absolutely, because it doesn't affect any you see, molecular mechanism, genomic or whatever, so it's also permanent. It's block the action of gene, but without any effects on its genes itself.
It could be a great idea if you do a combination. For example, people with brain tumors, some of them suffer from headaches, horrible headaches. So what they do surgery, so you have to open the skull. At the same time, you can introduce a pump with the medicine.
Yeah, we can just use the same pumps, but with such genetics, molecules. Yeah, thank you.
And the last speaker today will be Pavel, who will be talking again from the Phonology Institute of Physiology, who will be talking about the mysteries of joy.
Добрий день, hello everybody. Я попробую трошки розважити шановних гостей і жителів Львова чимось дуже відмінним від того, чим займаєтеся ви. Бо незважаючи на те, що я розумію, що ядро – це дуже важлива частина в функціонуванні, скажімо, нашого організму, але зрозуміло, що і підходи, які використовуємо ми, коли хочемо зрозуміти, яким чином нервова система обробляє інформацію, і взагалі експериментальні методики, які використовуються, повинні бути дещо різними. Нам особливо цікаво дивитися на організм тоді, коли він працює нормально, тому ми стараємося не використовувати ніякі методи розділення клітин до того, щоб досягти ядра, і не використовуємо методів, які є реально методами роботи з фіксованими препаратами. Хочемо робити з живими клітинами, і дуже бажано, щоб ці живі клітини взаємодіяли на протязі наших експериментів. І тому це викликає зовсім інші підходи до наших досліджень. Я трошечки просто розповім про деякі з цих підходів, те, як ми їх використовуємо, і надію, що це буде дуже цікаво, бо підходи трошечки інші. Ну, це план мого виступу, і я хочу сказати, що одна з головних проблем, яка виникає у нас, на відміну від тих, хто досліджує ядро, це взагалі побачити мозок. Переді мною дуже велика аудиторія, але я, на жаль, не можу побачити, що має місце в ваших мізках, тому що вони, на жаль, недоступні для мене, тому що вони скриті за допомогою тканин, і ці тканини, на жаль, не прозорі. З одного боку, це проблема, з другого боку, ми всі знаємо, що ми майже повністю складаємося з води, і вода є досить прозорою субстанцією, і тому, коли ми дивимося на клітини на рівні окремих клітин, то вони є досить прозорими і взагалі побачити навіть дуже важко. Тому у нас виникають дві проблеми. Перша проблема – ми не можемо добратися до мозку, а друга, коли ми добралися, ми не можемо побачити діючі молекули і клітини. Клітина, мабуть, є клітина, і місце контакту між клітинами синапс є для нас центральними елементами, які ми хочемо досліджувати, і ми хочемо досліджувати їх, звісно, у великій кількості. Тому головні методи, які ми використовуємо, це методи, в яких ми пробуємо помітити ті молекули, які для нас цікаві, і флуоресцентні підходи для цього є state of the art в нашому полі. І, наприклад, тут дуже гарно видно, що використання такого дуже, може, давнього, це перший флуоресцентний барвик, який був синтезований компанією Bayer більше 100 років назад. Це флуоресцин, і він був свого часу дуже цікаво використаний не для цілей досліджень в області мозку, а в області того, як з'єднуються річки у Європі. Було дуже цікаво для практичних цілей, чи Рейн з'єднується з Майном. І було зроблено дуже цікавий досвід. 10 кілограмів флуоресцину було вилито в одну річку, і після цього, оскільки флуоресценція є дуже чутливим методом, який дозволяє вимірювати, фіксувати концентрації 10 мінус 12 молі, то дуже швидко було видно, що через якісь системи протоків цей барвник попав в русло іншої річки. Тут так само, бачите, вода, яка є в цьому стакані, її структура зовсім неможливо побачити, бо вона прозора, але якщо ми додамо хоч трошечки флуоресцину, ми одразу бачимо цікаві моменти в тому, як ця вода переміщується. Так само ми робимо з біологічними молекулами. Білок є для нас та для всіх головною молекулою, різні білки, які досліджуються, і ми хочемо помітити їх певними флуоресцентними мітками, і зразу ми отримуємо багато більше інформації про те, як цей білок розташований. Якщо це жива функціонуюча клітина, то ми бачимо, як він переміщується при виконанні певних функцій, які нас цікавлять. Квінтесенцією цього підходу є підход, який ви всі добре знаєте, з застосуванням зелених, кольорових флуоресцентних протеїнів, які дозволяють за допомогою ефемерних підходів помітити практично будь-який білок з допомогою невеличкої лампочки, яка дозволяє нам відслідковувати, як цей білок синтезується, переміщується і працює в складі організму. І квінтесенція Proof of Concept – це тваринки, яких ви бачите, білок експресується в кожній клітині тіла. Звісно, це результати приблизно 20-річної давнини, але вони підштовхнули дуже сильно розвиток нейросайенсу. Це та область, в якій працюю я. І методологія нейросайенсу, вона досить з одного боку проста, вона така сама, як ми робимо, коли ми хочемо щось експериментувати або вияснити щось цікаве про якийсь механізм, наприклад, часи або автомобіль. Ми хочемо, звісно, розібрати його на частини, подивитися, як ці частини зв'язані один з одним, а потім попробувати зібрати, і може, на наше щастя, воно запрацює так само, як воно працювало до того. Можемо сказати, що взагалі це виходить. І тому зараз дуже поширена напрямок нейросайенсу, який називається конектосоміка. Українською мовою, так я її переклав. Це взагалі наука, яка пробує зрозуміти, як саме побудована нервова система на рівні окремих клітин і самих великих, можливо, субклітинних частин. Ну, є декілька підходів. Частина з них використовує зеленокресентні білки як можливість подивитися, як розподілення протеїнів, частина імуністохімію, частина інші підходи, але їх ідеологія така одна й та сама, береться певна частина мозку, і ця частина мозку нарізається на тоненькі слайси, в слайсах далі походить забарвлення, і після цього реконструюється на рівні оптичної мікроскопії, іноді з додаванням електронної мікроскопії, розподіл певних молекул. Зараз ми можемо дивитися, відслідковувати розподіл десятків молекул. Після цього все це реконструюється, і в результаті ми можемо побачити щось на кшталт. Ми можемо запустити це насправді мові. Це дуже невеличка частина мозку криси. Це мірна паличка в 15 мікронів. Це чудова морфологічна структура, яка називається дендритом, яка покрита вся невеличкими структурами, які називаються шипиками. І ми бачимо, що взагалі інші кольори визначають судини, гліальні клітини, інші частини нейронів, які розташовані тут. І ми бачимо, наскільки складно побудована навіть ця дуже-дуже маленька частинка мозку в розмірі порядка 15 мікрон. Але незважаючи на це, вчені надіються, що реконструюючи таким чином структуру мозку, вони зможуть зрозуміти, як мозок працює і обробляє інформацію. Це велика досить направлення було в цьому, і фінальною ціллю, я так розумію, для людства тут є побудова щось схожого на кшталт Nucleus, тільки для мозку. Пробуємо запустити. Це
Most people are familiar with the idea of Nucleus. This is a similar.
Тобто, взагалі, досить швидко розвивається проєкт по тому, як побудований мозок миші. Хочу вам зразу сказати, що дійсно є кількісні відмінності, які призводять до того, що виникають фантастичні відмінності вже в якості. Наприклад, простіше всього було вивчати маленького черв'ячка C. elegans, який дуже допоміг розумінню того, як побудована нервова система. Всі черв'ячки C. elegans однакові, кожного черв'ячка всього 302 нейрони. Ці нейрони зв'язані 5 тисячами синаптичних зв'язків, і що дуже цікаво, всі черв'ячки зовсім однакові. Тому ви можете проводити досліди не так, як з людьми або мишками. Ви можете бути певні, що все побудовано однаково. І ціною неймовірних зусиль, десь близько 50 років тому, вся ця досить складна система мозку цього черв'ячка була розшифрована. І для мене, наприклад, це було дуже важливим для подальшого розуміння того, що я буду робити в науці, бо після того, як вся структура була розшифрована, так ніхто і не розуміє, як саме цей черв'ячок виконує чимало дуже складних функцій на основі того, що структура його мозку саме така. Тому цей напрямок, незважаючи на те, що він фантастичний, дає нам змогу побачити мозок на рівні окремих синапсів, які з'єднують один з одним. Я не думаю, що він дійсно призведе до якоїсь революції, хоча це дуже важливий напрям досліджень в області досліджень мозку. Другий підход, який, можливо, ще хочу вас зацікавити цим підходом, бо цей підход дуже цікавий для мене, як для фізика. З одного боку, ми досить непрозорі, я дивлюсь на вас, ставлю руку, я вас не бачу, це приблизно 1 см моєї тканини. Але Я думаю, що це повинно бути дивно, особливо для хіміків, тому що ми теж розуміємо, що не це вода, і вода це прозора субстанція, тим не менше ми не прозорі. Непрозорість наша пов'язана саме з тими ліпідами, про які ми сьогодні багато казали. І тому, якщо ми зробимо простий досить біохімічний підхід, в якому ми зафіксуємо тканину, а після того приберемо ліпіди з цієї тканини, то тканина стає прозорою. Ось вам приклад, це мозок миші, який був просто зафіксований і лежить на певному написі, який якраз демонструє прозорість цього, непрозорість цього мозку. І після цього він оброблений за допомогою методики, яка взагалі носить назву методика прозорості. І ось тут лежить той самий мозок, і ми бачимо, що ми можемо читати, незважаючи на те, що в нас десь близько 3 сантиметрів тканини знаходиться між нами і цим написом. Такий підход дозволив, цей підход розвивається досить широко, але я думаю, що його квінтесенцією являється дослідження, які проводяться в Сполучених Штатах Америки. Я хочу вам показати красоту, яка виникає, коли цей підхід використовується у житті. Його додаткова позитивна сторона цього підходу є в тому, що connectivity, з'єднання нейронів один з одним, вони залишаються інтактними в такому препараті, на відміну від всіх інших підходів, коли ми з вами пробували нарізати тонкі зрізи і після того співставляти їх за допомогою спеціальних математичних процедур для того, щоб можна було відслідкувати, наприклад, ці абсолютно фантастичні дендритні дерева цих аксонів. Це кора мозку криси, а це підкоркові вже структури. Ось. І насправді я думаю, що дійсно це питання, можливо, десятиліття або 2 десятиліть, і ми дійсно зможемо реконструювати повністю структурну організацію мозку людей. Я майже впевнений, що результати будуть точно такі ж, як результати проєкту «Геном людини», коли ми думали, що ми скануємо геном, і після того ми вилікуємо всі хвороби, бо ми знаємо тепер про геном все. Зрозуміло, що ця ціль не була досягнута, але з другого боку це привело к фантастичним можливостям для наших досліджень. Без цього проєкту, я думаю, людство не могло продвигатися так швидко. Тому я думаю, що якимсь чином це буде теж позитивно для людства і для нейросайенсу, в якому я працюю. Бачите, що тут можна відслідковувати окремі аксони, це структура, яка приблизно 1 мікрон товщину на багато сотень міліметрів, на багато метрів, або навіть на сантиметр. Але, мабуть, більш цікаво все-таки розпізнати не тільки структуру, а й побачити, як ця структура працює. Знову ж таки, ми стикаємося з проблемами розмірності, як я вже сказав. Черв'ячок Сі елеганс має 402 нейрони, рибка, яку показувала Нана, має приблизно 75 тисяч нейронів у своєму складі свого мозку. Миша десь 75 мільйонів, а ми близько 100 мільярдів клітин, які з'єднані приблизно 100 трильйонами синаптичних контактів. Це фантастично складна система, і я більше звертаюся до молодих людей. Зрозуміло, що підходи, які хочуть реконструювати усе після того, зрозуміти, як працює мозок, мені здається, при такій складності системи не будуть реалізовані, і треба придумати, в принципі, якісь нові підходи, такі як свого часу фізики придумали для того, щоб описувати газові середовища за допомогою 2-3 параметрів. Так само треба придумати щось для цієї складної системи нашого мозку. І я думаю, що це повинні робити люди, які працюють на стику між математикою і біологією. І я думаю, що ця сфера діяльності буде зараз дуже перспективна. Але одне діло зрозуміти, але я думаю, що практичні інтереси, які стоять перед людством, це не просто зрозуміти. Для мене, наприклад, цікавіше за все зрозуміти, як побудована система. Але людство хоче не цього, я думаю, в першу чергу воно хоче практичних наслідків, а саме вилікувати невиліковні хвороби, які, на жаль, у нас є. І, на мою думку. Ми сьогодні розглядали підходи імунологічні, які дозволяють контролювати такі процеси, наприклад, як розвиток раку. Але я думаю, що над всім стоїть такий мозок. І якщо ми навчимося за допомогою мозку правильно контролювати імунну систему, то це буде найбільш адекватний, правильний спосіб лікування абсолютно всіх хвороб. Тому я хочу, щоб більше людей прийшло до дослідження мозку, і, можливо, це дозволить людству через якийсь час добитися того, що ми вилікуємо більшу частину хвороб, які нас зараз торкаються. Ну, я просто хочу сказати, що зараз ми досить широко використовуємо підходи, які основані на досить новій галузі досліджень, називається генетика. Це галузі, які ми за допомогою молекул, які ми експресуємо у певних клітинах або групах клітин за своїм бажанням, як правило, використовуючи генетичні підходи або вірусні конструкти, ми можемо управляти певними групами клітин за допомогою світла, так як ми хочемо. І це дає можливості для дуже широких досліджень, які, можливо, приведуть, по-перше, до кращого розуміння, а по-друге, до можливості управляти мозоком за допомогою світла. Можливо, це такий магістральний шлях для вирішення багатьох проблем, зв'язаних з здоров'ям. Ну, взагалі, сам підход заснований на, він з'явився десь близько, трошки менше 20 років назад, і він заснований на зовсім простих ідеях, які я дивуюсь, що людство не привело до досліджень раніше. Це археобактерія, яка росте на чашці Петрі. Це одноклітинне створіння, і тим не менш, коли освітлюєте цю чашку Петрі, вона збирається в темному кутку. Зрозуміло, що люди, які задали собі питання, чому так, вони і були тими дослідниками, які знайшли молекулу, яка є білковою молекулою, що є з одного боку сенсором світла, а з другого боку компонентом електронних мереж в мембрані клітин. І це дозволяє за допомогою цієї молекули, яка не експресується в клітинах в період, це дозволяє взагалі робити фантастично цікаві досліди, які я вам покажу пару прикладів, які ми в своїй лабораторії отримали. Одна з задач, яка в нас є, ми співпрацюємо з нами в певних проєктах, це дослідження і механізмів болю, і способів подолання болю. І, звісно, якщо в нас є периферійні закінчення в структурі тіла нашого організму, то ми можемо їх за допомогою підходів, які я вам сказав, або збуджувати, або гальмувати. Збудження дозволяє нам проводити дослідження певних типів клітин, а гальмування – це прямий спосіб лікування болю. Якщо ви за допомогою простої процедури типу ін'єкції вірусних конструктів, ту ділянку периферії нашого тіла, де ви відчуваєте біль, хронічний біль, який турбує людину на протязі днів, неділь, місяців, ви робите ін'єкцію, після цього ви можете підторможувати ті нейрони, які сповіщають про цей біль, просто прикладаючи до них світло в певної довжини хвилі, і тим самим вибухуєте. І ці підходи зараз розвиваються, є вже декілька компаній, стартапів, які працюють над реалізацією цієї ідеї вже в лікувальній процедурі. А тут у нас, ну я покажу, ми, наприклад, працюємо з тими нервовими шляхами, які забезпечують відчуття тепла. І одночасно біль, який пов'язаний з тим, що ви прикладаєте щось дуже гаряче до периферійних тканин організму. Ну, не думаю, що тут дуже гарно видно, але хочу сказати, що це система проводів, яка доводить, сигнал про біль з периферії до спинного мозку. Саме це у нас спинний мозок, а це те, що називається dorsal roots, не знаю, як це буде українською мовою, нейронні корінці, так, бажаємо Англії, які попадають і приносять інформацію про те, що трапляється на периферії з температурною чутливістю. І навіть якщо ми візьмемо такий маленький шматочок, який показаний тут білим, ми бачимо, наскільки складна ця система проводів, яка забезпечує необхідні відчуття. І для її дослідження ми пробуємо робити і тваринні моделі. І ось давайте запустимо цей фільм, я думаю, що я покажу. Ось ми експресували за допомогою вірусного. Ні, це генетично модифіковані миші, але ми працюємо з вірусними конструктами з крисами. Ми в одну з лапок, здається, завжди в ліву лапу, експресуємо конструкти, які активуються світлом саме в тих нервових закінченнях, які відповідають на тепло. І в результаті ми отримуємо, бачите, можливість, якщо ми посвітимо світлом на лапу такої тварини, їй дуже боляче. Це ін'єкція була зроблена, верніше, генетична модифікація була зроблена в цій лапці, а тепер ми посвітимо лапку, яка, чи ми вже пропустили на протязі. Ну так, оце ми світимо в контралатеральну лапку, яка не була задіяна. Це просто луч лазера, як луч був український, промінь лазера, і ніякої реакції немає. Але тільки ви світите на ту лапку, в якій експресується даний конструкт, тваринка відчуває наші облік. І швидко прибирає цю лапку, починає її лизати. Бачите, це лапка контралатеральна, нічого. А ось лапка, і вона зразу, бачите, вона миттєво прибирає, це дуже боляче. Ось, таким чином ми маємо моделі, які ми знаємо, напевне, що ми експресували правильний конструкт саме тих клітин, які треба, і далі ми працюємо за допомогою досить складних електрофізіологічних іміджингових підходів і пробуємо зрозуміти, як ота складна система проводів, які я вам показував, працює, забезпечує відчуття гострого болю при активації периферійних теплових рецепторів. Дякую за увагу. Мене пробачить, це моя перша лекція в житті українською мовою.
So, I would like to thank, other questions? Questions?
I'm really scared, but it's a very interesting talk. Thank you very much. I wonder if, I don't know whether you have this technology of CRISPR-Cas non-genome editing to, enable in vivo genes with color proteins, because they have shown very nice optogenetics experiments where they're planning to use it.
Actually, we don't use, and basically in our field now it is considered that the Krellox technology is much better. And second, I personally prefer to use viral constructs, because in this case you can have local expression and at the time which you would like to have this expression. And this is the main methodological approach which we use in Genome. I think that
No more questions? Thank you very much. So, on behalf of Roman and me, I would like to thank you all for attending the meeting. I would like to thank the speakers for an excellent presentation and for the effort they made to do it as understandable as possible to the general audience. And before finishing, I would just to tell you, to remind you why we are here. We are here because we are organizing a meeting. And actually, what you need to know about the meeting, if you want to attend, if you want to participate, if you want to present your data, do not hesitate. And actually, the address, just the only thing you need to know is the address, which is here it is. And I invite you to visit the site, to subscribe to the news of the site. We will plan to have some fellowships for young people who want to attend and who want to present their data. So do not hesitate to apply and to visit the meeting. It will be held in June. It will be held in June 2020. Here, I believe, yes. So thank you all, and hopefully see you next year.